So we have a busy schedule today. So I briefly summarize what we had yesterday. So Kerry Chang, I was really like a talk and kind of refresh my knowledge about the ML physics and also the make me much more appreciate what the new machine will bring. And so it was great, and followed by Jong-Ho Lee. And actually, Jong-Ho Lee organized the whole this symposium and closely worked with Kerry Chang and other invited speakers, including Seung Che and Fukunaga. Fukunaga's introduction of the NIPS is correct way? Yeah, it's amazing center. So it's almost like a NIH, the imaging facility. You have 70 and 3D and all different animal imaging. And also, the even more exciting was that was synchronized scanning. It was really impressive. And Jiuk Cha kind of, in my opinion, gave a provocative talk. Made me think a lot about what AI can do for us, but also how wisely we can use that technique and how, bring me a lot of thoughts about that. I think you're on the same page with me. All the students given huge interest in this artificial intelligence, the surge in this age. And also, it was nice, the way of ending yesterday's talk. This is sum up and just talk about the new machine we're getting. And actually, after the first two talks today, and the speakers and the all core faculty members will go to the center, and then we have inauguration ceremony. So that's why I dressed up like this. So please understand my outfit today. So I also learned a lot from his presentation, which was quite different from what I got last year. So that means a lot of ML researchers start buying this machine and apply the machine to various purposes. And so that give us the lesson that we have to really think carefully how wisely use that machine and combine with all other different topics and the way of approaching the brain science. So, and also I briefly introduced today's session. This is a session 2, second day. And this is a little bit different from what he had yesterday. This is more on content. Yesterday, although yesterday talks, of course, included a lot of interesting topics in cognitive neuroscience and clinical neuroscience, but today specifically focused on this, the translational research, mental health, and value-based perceptual decision making, including my talk. But also I have to give you a sad news that Eli couldn't make it here today because he's working at NIH, so government employee. So you know that a lot of stuff's going on in the US. So it's a government shutdown. So it's illegal for him to give any talk. So he cannot give even Zoom talk. So, but I I begged him to still posting his name in this symposium poster. That was allowed, granted. So, but anyway, I really missing his talk, and so I I feel sorry for that. So, are you ready to? Yeah. So the Professor Yun Sang Lee is one of the research scientists at the center. He did a great job as moderator today, and today he'll continue to do service for us. Please.
Good morning, everyone. Happy Halloween. So this is the second day of the symposium, and I'm happy to introduce Dr. Shinichiro Luke Nakajima. And he's going to give a presentation titled Dopamine and Glutamate Hypotheses for Treatment Response in Schizophrenia.
Thank you very much for inviting me. So especially for Professor Lee and Professor Kim. So today I'd like to talk about dopamine and glutamate hypothesis for treatment-resistant treatment response in schizophrenia. So first of all, I will talk about current issues in psychiatry. The prevalence rate of mental illness is about 20 to 30%. And a third of people with mental illness do not respond to currently available treatment. And no treatment available for cognitive impairment or suicidality, which I got to know yesterday. This is also a problem of society in Korea. And it is also a big problem in Japan. And pathophysiology remains unclear of mental illness, despite a host of research, which is maybe attributable to clinical or biological heterogeneity and comorbidity. And as a result, pharmaceutical companies have almost stopped the development of novel treatment. Therefore, there is need for interdisciplinary solution for social issues of psychiatry. So such an effort of SNUBIC. One of the issues around psychiatry is shorter life expectancy. This must be surprising. And so please look at this figure, a bit dirty figure. So the upper figure is about males with mental illness, and lower figure is females with mental illness. And just y-axis is how long they pass away. So it represents from about 5 to 20 years. For example, schizophrenia, so 15 years. And it is usually regarded as because of suicidality or sudden death. But actually, it's also due to cardiorespiratory diseases. Have you ever read this book? This is a New York Times bestseller. And this book is about one family, including maybe more than 12 family members with the third generation, but most of them have schizophrenia or schizophrenia spectrum disorders. And the story is about, of course, suffering of this family and the recovery of this family, and also about the academic career of young scientists, especially focusing on genetics. And on other note, this book describes the treatment in the 80s, but which is not so different from currently available treatment. Therefore, I believe I need to continue to research as clinician scientist. So to address these social issues related to psychiatry, I have been doing clinical research with my research team. So today's talk is all what they have done. So let's go to the main contents. So the first one is glutamate dopamine hypothesis of schizophrenia. And the second one is MRS, so proton magnetic resonance spectroscopy study on treatment-resistant schizophrenia. And finally, I'd like to address limitations and future directions. At first, I'd like to introduce glutamate hypothesis of schizophrenia. NMDA receptor antagonists, such as ketamine, cause positive and negative symptoms and cognitive impairment in healthy controls. NMDA receptor antagonists exacerbate these symptoms in schizophrenia even during taking antipsychotics, so dopamine receptor antagonists. In addition, anti-NMDA receptor encephalitis is known to cause psychotic symptoms. Furthermore, so genome-wide association studies on schizophrenia noted several genes encode proteins involved in glutamatergic neurotransmission. Therefore, these results suggest NMDA receptor hyperfunction or glutamatergic dysfunction may have a link to etiology of schizophrenia. So this is a summary of a meta-analysis to examine glutamatergic or GABAergic metabolites measured with MRS in patients with schizophrenia. So please look at basal ganglia. In the basal ganglia, glutamatergic neurometabolites are increased in patients with schizophrenia spectrum disorders. Here, schizophrenia spectrum includes clinical high risk for psychosis or first episode psychosis and acutely ill or chronically ill patient with schizophrenia. So in this spectrum, as a whole, glutamatergic metabolites are increased, and these elevations are normalized with antipsychotic treatment or illness course. So please look at mid-cingulate cortex. In contrast, glutamatergic metabolites and GABA levels are decreased, and these decreases emerged after onset of schizophrenia, which means so conversion from clinical high risk of psychosis to first episode psychosis. One of our colleagues developed novel PET tracer to visualize AMPA receptor. It is a important molecule in the glutamatergic system. And using this tracer, we examined AMPA receptor density distribution in patients with schizophrenia. We found decreases in AMPA receptor density in patients with schizophrenia, including the areas of hippocampus or basal ganglia and cingulate cortex. We also found negative associations between AMPA receptor density and clinical symptoms. These results also support glutamatergic hypothesis of schizophrenia. So let's go on to dopamine hypothesis. So dopamine PET studies have reported dopamine, so presynaptic dopamine is increased in the striatum of patients with schizophrenia, including dopamine synthesis capacity and dopamine release capacity and occupancy of endogenous dopamine in the synapse vesicle. Also, using MRI, we can measure dopamine function, especially measuring neuromelanin. Neuromelanin is a biomarker for mid-brain dopamine neuron function. At first, we conducted a meta-analysis of neuromelanin MRI studies and also post-mortem studies, comparing neuromelanin levels between patients with schizophrenia and healthy controls. 5 studies employed neuromelanin-sensitive MRI and 2 were post-mortem brain studies. We found the patient group, schizophrenia group, showed higher neuromelanin levels in the substance Niagara in comparison with healthy controls with moderate effect size. So these results also corroborate dopamine hypothesis of schizophrenia. So in summary, PET and neuromelanin MRI studies have reported presynaptic dopamine function is elevated from so substance Niagara to striatum. So this is the nigrostriatal pathway. And this result supports the dopamine hypothesis of schizophrenia. Please look at the right figure. Now we are able to bridge the glutamate hypothesis and dopamine hypothesis of schizophrenia. So glutamatergic dysfunction is found mainly in the medial prefrontal cortex and basal ganglia. And dopamine dysfunction is detected mainly in the midbrain and striatum. And these dysfunctions are connected via EI, excitatory and inhibitory imbalance in the subcortical regions. So let's go on to move on to MRI studies on treatment-resistant schizophrenia. So why treatment-resistant schizophrenia is important? 30% of patients do not respond to antipsychotics. So antipsychotics means dopamine receptor antagonist or dopamine receptor partial agonist. And most patients with TRS, so treatment-resistant schizophrenia, don't respond to first-line antipsychotics, even from illness onset. So these findings suggest treatment-resistant schizophrenia, TRS, may be a distinct category among patients with schizophrenia. We conducted a systematic review about 10 years ago, and we found lack of neuroimaging correlates of TRS. At that time, 25 neuroimaging studies on TRS were published, and there were no replicated neuroimaging findings specific to TRS. So therefore, this review suggested further research would be needed to examine glutamatergic dysfunction, mainly glutamatergic dysfunction in patients with TRS. So I'd like to introduce several MRS or dopamine PET studies focusing on TRS. So please look at left figure. So they compared dopamine synthesis capacity measured with PET, and so among healthy controls and TRS and responders, non-TRS. They found dopamine synthesis capacity is higher in responders, but not in TRS. The same group conducted similar design study, and again they found dopamine synthesis capacity is higher in non-TRS, responders, and dopamine synthesis capacity is not different between patients with TRS and healthy controls. So these results suggest dopamine synthesis capacity is lower in patients with TRS in comparison with non-TRS after antipsychotic treatment. So this is great study, fabulous study conducted by Professor Kim, and they compared dopamine synthesis capacity among healthy controls and first-line antipsychotic responders and clozapine responders. And again, they found dopamine synthesis capacity is lower in patients with TRS on clozapine in comparison with other groups after antipsychotic treatment. Oh, by the way, clozapine is the only antipsychotic indicated for TRS. So let's go to MRI studies. Please look at left figure. So they compared glutamate levels in the anterior cingulate cortex among healthy controls and treatment responders and TRS. And in contrast to dopamine PET studies, they found ACC glutamate levels are increased in TRS in comparison with healthy controls. They didn't find any significant differences in ACC glutamate levels between responders and healthy controls. The same group reported similar findings. ACC glutamate levels is higher in TRS in comparison with responders. However, other group reported now findings on clozapine response and ACC glutamate levels in patients with schizophrenia. So this figure depicts the difference in glutamate system in anterior cingulate cortex and mid cingulate cortex. Red is mid cingulate cortex, so MCC, and blue is anterior cingulate cortex, ACC. So MCC is mainly related to cognition, including decision making, and rich with NMDA receptors, relative to AMPA receptors. And ACC is mainly associated with emotion and rich with AMPA receptors. In healthy controls, glutamate levels are higher in the ACC in comparison with MCC. And as I mentioned before, glutamatergic metabolite levels are lower in the MCC of schizophrenia. In contrast, there is no difference in the ACC glutamate levels in patients with schizophrenia. Therefore, the previous studies only focused on ACC, such that I'd like to start studies focusing on MCC glutamate levels in a patient with schizophrenia. So from now on, I'd like to introduce two studies from our group that examined glutamate levels in MCC of patients with TRS. We compared MCC glutamate levels among patients with TRS and patients with non-TRS, also healthy controls. We included 95 participants, 29 TRS, 33 non-TRS, and 33 healthy controls, age and sex matched. And we found glutamate levels in the MCC were higher in a patient with TRS in comparison with healthy controls, while no group differences were found in the caudate glutamate levels among the groups. So this is another study. We compared MCC glutamate levels among healthy controls, patients who responded to first-line antipsychotics, and patients with TRS who responded to clozapine, non-URS, non-alcohol-resistant schizophrenia, and patients who did not respond to clozapine, patients with alcohol-resistant schizophrenia. We included 100 participants, including 26 alcohol-resistant schizophrenia, 27 non-alcohol-resistant schizophrenia, 21 non-TRS, first-line antipsychotic responders, and 26 healthy controls. As a result, there's higher MCC glutamate levels in patients with clozapine non-responders in comparison with healthy controls. On the other hand, in the caudate and DLPFC, there are no group differences in glutamate levels. So, as a summary, these findings suggest higher levels of glutamatergic metabolites in the ACC and MCC may be among the shared biological characteristics of treatment resistance to antipsychotics, not only first-line antipsychotics, but also clozapine. So again, in the meta-analysis I mentioned before, MCC glutamate levels were lower in patients with schizophrenia, but mainly which attributable to treatment responders. In patients with TRS, there is evidence, including our studies, so higher glutamate levels or glutamate levels in the MCC were detected. So these results suggest while patients with schizophrenia generally have lower glutamatergic levels in the MCC, a certain subgroup, mainly TRS, may have higher glutamatergic metabolite levels in this region, and which may contribute to treatment-resistant antipsychotics, including clozapine. On other note, we examined GABAergic levels in the MCC in patients with schizophrenia. In order to examine the relationship between clozapine response and MCC GABA levels. In general, GABA levels are decreased in the MCC of patients with schizophrenia. Also, clozapine may have exerted its effect via a GABAergic system. We included 100 participants, including 21 clozapine responders with TRS, 22 clozapine non-responders with TRS, and 15 non-clozapine antipsychotic responders, and 33 healthy controls. We found higher MCC GABA levels in clozapine non-responders. So let's go to limitations of these studies, and I'd like to show future directions of this field. As you see, these figures, we found statistical group differences in glutamatergic or GABAergic levels among the groups. That's why these studies were published. But as a self-criticism, for example, please look at this person with TRS. So this person with TRS showed lower glutamate levels compared with healthy controls, which is opposite of my findings of this paper. So thus we cannot use glutamate levels as a biomarker in the ordinary clinical settings like CRP as a measure of information, because there are huge variations in each group. In addition, for example, this study is one of the largest studies conducted in UK. This study aimed at comparing ACC glutamate levels and striatal presynaptic dopamine synthesis capacity between antipsychotic responders and non-responders. They found ACC glutamate levels a bit higher in responders relative to non-responders, relative to responders, but effect size is very small. And also the accuracy of the ACC glutamate levels for predicting antipsychotic response is very low. Moreover, they didn't find a group difference in dopamine synthesis capacity even with a largest sample. This is, in my opinion, the limitations of conventional approach for clinical psychiatric field. Previous studies analyzed univariate analysis correlations of clinical and biological variables within the same disease, or have compared biological variables between the groups. They fail to address clinical or biological heterogeneity and much morbidity in psychiatric diseases. And statistical significance in conventional approach do not lead to the elucidation of pathophysiology or reconstruction of diagnosis of mental illness. So I told dopamine synthesis capacity is higher in responders compared with non-responders or healthy controls. But please focus on responders. So please, at first, please look at upper left figure. In responders, dopamine synthesis capacity is higher. And please look at so lower left figure. Dopamine synthesis capacity is not different between so first line responders and healthy controls. And please look at right figure. So this is about remission. Remission means so loss of positive symptoms. So complete, in a sense, so before complete full dissolution of symptoms. So in patient with remission, dopamine synthesis capacity is lower. So these findings suggest so maybe inconsistent findings on dopamine synthesis capacity in relation to antipsychotics may exist in patients with schizophrenia. And also, I'd like to introduce our neuromelanin MRI studies. So in this slide, so please look at left figure. We compared neuromelanin levels in the substance Niagara between patient with TRS and patient with responders and healthy controls. We found lower neuromelanin levels in the ventral substance Niagara of TRS, and ventral substance Niagara is included by nigrostriatal pathway mainly, which is consistent with dopamine hypothesis. However, please look at right figure. We again compared in a different institution, neuromelanin levels in the substance Niagara among patients with responders, non-responders, and healthy controls. In contrast to study depicted in left figure, we found higher neuromelanin levels in the dorsal substance Niagara in patients with TRS. Dorsal substance Niagara contributes both mesolimbic and nigrostriatal pathways, and with a relatively greater role in the mesolimbic pathway. Thus, these findings suggest dopamine dysfunction in this pathway may play different roles in treatment responsiveness in patients with schizophrenia. So from now on, I'd like to explain about biological or clinical heterogeneity or morbidity in patients with mental illness. So please look at this figure. X-axis is dopamine synthesis capacity in the striatum, and Y-axis is glutamate levels in the cingulate cortex. And red line depicts schizophrenia, and blue line depicts healthy controls. Based on meta-analysis, we found dopamine synthesis capacity is higher, and ACC glutamate levels are lower. So patients with schizophrenia should exist in this area. But we also find a subgroup of patients with schizophrenia here. So lower dopamine synthesis capacity and higher glutamate levels. In addition, please look at this area. There are no significant differences in ACC glutamate levels or dopamine synthesis capacity in the striatum between patients with schizophrenia and healthy controls. These results suggest even with famous dopamine hypothesis or glutamate hypothesis cannot fully explain that the picture of or etiology of schizophrenia. So this may be one of the limitations of current neuroimaging research related to schizophrenia. Then, I'd like to talk about clinical heterogeneity in relation to diagnosis. Clinicians make diagnosis of psychiatric disorders by clinical interviews with patients. Just spending 1 hour or 2 hours talking with patients. And diagnosis were based on symptom items of each disorder. And the same diagnosis have different presentations in definition. For example, in order to fulfill the diagnosis of depression, we need to detect 5 items among 8 symptoms. But for example, in the item of sleep, it may include lack of sleep or hypersleep. And also, for example, in appetite, it includes hyperappetite or lack of appetite in the diagnosis of depression. So that's why the kinds of depression is higher than 1,000. So let's go on to clinical heterogeneity in terms of treatment or response. As I mentioned, 1/3 of patients do not respond to psychotropics, not only schizophrenia, but also depression or bipolar disorder. But most of the treatment are monoaminergic or glutamatergic. So therefore, I assume treatment response may be related to the etiology, so heterogeneous etiology of psychiatric disorders. Biological heterogeneity also exists in structure of mental illness. In general, it is reported brain volumes are reduced in certain area of schizophrenia. So maybe you can imagine this figure. In fact, these lower 2 figures represent a true picture of brain hypertrophy or hyperatrophy or hypertrophy of patient with schizophrenia. There are a few areas with consistently reduced volumes or increased volumes within patient with schizophrenia. And consistency means just 2 to 5% among patient populations. So let's go to clinical match morbidity. Clinical match morbidity is very common. So please look at orange bar. This says third disorder group, including schizophrenia. And more than 90% of patients with third disorder have other comorbid disorders. For example, externalizing or internalizing disorders, including depression or anxiety disorders. So this figure technically suggests 70% of mental illness have comorbid disorders, which suggests much morbidity. Clinical match morbidity is also exists in treatment response. So in the clinical settings, the same treatment are effective to different disorders. Of course, antipsychotics, dopamine receptor antagonists are effective to schizophrenia, but which is also indicated for bipolar disorders. And with a small lower dose of antipsychotics, it is also effective to depression. On other note, other example, epileptics are effective to, of course, epilepsy as well as bipolar disorders. Also, please imagine psychotherapy. Famous one is CBT, cognitive behavioral therapy. CBT is mainly used for depression or anxiety disorders, but it is also useful for other mental illness. These findings suggest given that treatment option, one treatment option have one specific mechanism, but they are effective to different mental illness, which suggests different diagnoses may have shared mechanisms. So let's go to genetics. So there's a substantial genetic overlap based on GWAS studies. For example, between schizophrenia and bipolar disorder, or anorexia nervosa and OCD. So today I talked about only the dopamine and glutamate hypothesis. So glutamate and dopamine, but many other factors have contributed to the etiology of schizophrenia. As such, today's presentation is a kind of blind man elephant situation. So we need to examine many different factors related to schizophrenia as a collaboration internationally. So as such, we need to switch from conventional approach with shallow phenotyping to multivariate and data-driven techniques, including deep phenotyping, and as a result, biotyping. So this is design of deep phenotyping cohort. So we have been building a deep phenotyping cohort, not only our group, but also other group in the world. So the aim is to solve the heterogeneity and much morbidity of mental illness. At first, we need to collect clinical data and biological data as much as possible from each subject with different mental illness. So here. And after the data collection, we do maybe machine learning or multivariate analysis for biotyping. So if we can find biotype, specific biotype, and then we'll be able to tackle on finding specific mechanisms for each biotype. And if we find specific mechanism related to each biotype, we can tackle on developing novel treatment. So if we can find circuit dysfunction in a specific biotype, then we'll be able to develop neuromodulation to improve neural dysfunction. Circuit dysfunction. So from now on, let me introduce several studies from our group using multivariate analysis or machine learning, based on deep phenotyping data. But please pay attention. I'm just a clinician scientist, and I don't have expertise of deep machine learning or multivariate analysis. So as a joke, just joke, but after my presentation, please don't ask me very strict questions about them. This is basically based on histology studies, they have reported ACC, cingulate cortex have several subregions, and which which play different roles in cognition. And using non-negative matrix factorization, we found and based on cortical thickness, surface area, and MD or FA values of ACC and MCC, we found 4 subregions in the data from Tokyo cohort. This is 4 subregions, and using Toronto cohort, we found again same 4 subregions. In the component 1 and 2, in the component 1, purple, and component 2 in the blue, this area, mainly mid cingulate cortex. Cortical thickness or MD in the mid cingulate cortex, component 1 and component 2 were different among treatment responders and treatment non-responders. And cortical thickness was decreased in the component 1 and 2 of patient with TRS, and MD was increased in a similar fashion. So these alterations suggest increased synaptic elimination and sparse axonal organization in these components. So in the MCC, in patient with treatment-resistant schizophrenia. Also, we examine the relationship between MCC glutamate levels and decreased cortical thickness or increased MD in component 1 and 2, so MCC. And we found decreased CT cortical thickness and increased MD in component 1 and 2 were related to MCC glutamate levels. So these results suggest glutamatergic dysfunction in the MCC may lead to structural alterations in inhibitory control-related subdivisions of MCC. I'd like to talk about several studies, several more studies applying machine learning to our MRI data. So the problem of neuroimaging studies is mostly cross-section. So we don't know which stage patients exist, so illness course or illness stage. So that's why we applied machine learning called SUSTAIN. So SUSTAIN is data-driven machine learning for subtype classification and staging from patients of brain morphological abnormalities, especially in a prospective fashion. So usually, they applied SUSTAIN in neurodegenerative disorders such as FTD or Alzheimer's diseases or ALS. So this is the first study to adapt SUSTAIN to patients with mental disorders. So in detail, we don't know. So for example, in this subject, we don't know. For example, imagine there are two trajectories of subtypes of schizophrenia, but we don't know each subject exists here or here. So we found three subtype trajectories of patients with schizophrenia. The first one is subcortical group, and the second one is globus pallidus increase initially, then cortical thinning follows, and the third one, cortical thinning mainly occurs. We examined the relationship between treatment response and subgroup detected by SUSTAIN, but we couldn't find any significant differences in the distribution of TRS. However, TRS is more progressive stages in each group. Also, globus pallidus type was associated with less stage progression, which is consistent with a previous report. So globus pallidus hypertrophy is related to good treatment response in patients with schizophrenia. We also try to link neuroimaging subtype and treatment response or other biomarkers. So in collaboration, international collaboration, including Seoul National University, thanks to Professor Kim, we successfully collected more than 500 patient schizophrenia data. We at first conducted cluster analysis using hierarchical clustering, and then we try to relate structure subtype and treatment response or structure subtype and other biomarkers. So this is an interim analysis. But let me show you preliminary result. We found three subtypes. Cortical atrophy type and subcortical atrophy type and global hypertrophy type. And as for the relationship with treatment response, there were significant associations between subtypes and treatment response. So hypertrophy type is related to good treatment response. So this is consistent with other report from our group or other group. So the hypertrophy of especially growth pallidus may be related to good treatment response in patients with schizophrenia. We also try to associate structure subtype and other biomarkers, including glutamate levels. ACC glutamate levels are higher in subtype 1 and 2, which is consistent with studies reporting glutamate levels are higher in patients with TRS. In contrast, substance Niagara neuromelanin levels are elevated in subtype 2. So subtype 2 is subcortical atrophy type, which is not associated with, which is not consistent with previous studies. So in order to consider time, so let's skip several studies. So here, yeah, I need to talk. So let me introduce structure covariance study. So structure covariance is a method for discovering networks of brain regions where coordinate cortical thickness changes are occurring based on similar pathophysiology, supported by fMRI or DTI. And in order, by using structure covariant method in patients with TRS, we found increased structure covariance in not TRS, centered in the insular or temporal gyrus. If you can find core regions of the network, abnormal network in patients with, for example, TRS, and then we may be able to develop neuromodulation treatment for difficult to treat condition. This is a region network mapping studies. So despite the mechanism pathophysiology underpinning similar underpinning same diagnosis, there are similar network, there are same network dysfunction based on region network mapping studies. So for example, as for depression, depression is induced by psychological stress or organic brain diseases or neurological disorders. So the etiology of depression are various, but despite the different etiologies, the depression network abnormality is shared. And the core of the depression network is the DLPFC. And the DLPFC is a target of transcranial magnetic stimulation treatment. And recent study reported, using region network mapping, the core of schizophrenia network is hippocampus. Therefore, we may be able to develop deep brain stimulation using TMS or other neuromodulation to target on hippocampus for the treatment of schizophrenia. So this is the final slide. Now we have been our collaboration has been tau network mapping and chemogenetic studies for the development of circuit treatment, or especially focusing on apathy. So first, we measure tau distribution with tau PET in patient with PSP, progressive supranuclear palsy, using tau PET. We found different tau distribution related to the same symptom. Here, apathy. Then, adapting region network mapping, we conducted tau network mapping and found the core of apathy circuit may be inferior frontal gyrus, a sulcus. And without directly going to TMS, we now are doing animal research using chemogenetics to examine the causality between this network and apathy. So if we validate this causality, we will start a clinical trial to examine the TMS on the core of the network on apathy. So in conclusion, patients with TRS have higher glutamatosic metabolite levels in the MCC, while glutamatosic metabolite levels in the MCC may be lower in the whole patient population, especially during early stage. The relationship between presynaptic dopamine function and treatment response may still remain unclear. Deep phenotyping and multivariate approach are warranted to address the clinical and biological heterogeneity and much morbidity of mental disorders. So thank you very much for your attention.
Thanks for the very interesting talk. We have some time for questions, and just a reminder that don't ask hard questions about machine learning. Any questions?
Thank you very much for your great talk. You mentioned neuromelanin imaging as a new methodology for examining dopamine system. And so I'm wondering how long the status of dopamine system is the neuromelanin imaging reflecting? Is it short or how long?
Thank you very much for your question. So neuromelanin represents the accumulation of dopamine metabolisms, like garbage disposition center in the neurons. So to avoid hypertoxicity of dopamine neurometabolisms. So it is reported neuromelanin is an index of long-term dopaminergic function, mainly at least maybe several years.
Any other questions? Please.
Hello. Thank you so much for your talk. One thing that I was kind of curious about is because symptoms and the diagnosis criteria for schizophrenia can differ depending on the country. I know that some countries use DSM, other countries use, I think it was ICD or something. Do you think that these kind of cultural differences, because you also acknowledge you use studies from Japan, you use studies from the UK. I was wondering if you ever encountered any problems from these cultural disparities, and if you did, how you dealt with them.
Thank you very much. For your question. So I agree with you. So the ICD and DSM are different. Maybe ICD is usually used for actual reimbursement of patient payment, and the DSM is mainly used for research. And when we use DSM for research, we usually semi-structured interview for DSM, but DSM has a major problem. It doesn't have it doesn't have how can I say, longitudinal history or illness progression of mental illness. This is one of the problems, and the other one is it's not related to conventional, traditional diagnosis of mental illness. For example, there are two peaks of onset in patient with schizophrenia. So early onset and late onset. And the phenomenon of these two populations are quite different, but which is not explained by the DSM. And also DSM doesn't include treatment response or neuroimaging. So so that's why, so, in a clinician's perspective, it's a bit frustrating. But so to tackle on this problem, at first, so we may need to take a distance from clinical symptoms because which may influence by so patients mentioning or ability or capacity of clinicians. So that's why, so when we analyze data, we may avoid using clinical variables and then just focus on biological data. So this is current my opinion.
Thank you very much. So I just want to follow up on this picture. I think what's actually showing, oh, the one you're showing, yeah, the one on the left. What are the color?
I see. So this is just corrected and uncorrected by neuroimaging analysis. Sorry for confusing. So please look at red line.
The red one. Let's look at the red one. And then is this suggesting a differences in different populations, or they are consistent between Toronto population and the Tokyo cohort?
Yeah, so this is these two studies employed the same design. So to compare dopamine function in the substance Niagara among TRS and non-TRS and healthy controls responders and non-responders. So same design, but as you know, neuroimaging, it is very difficult to replicate neuroimaging studies on mental disorders. So that's why I usually do 3 studies with same design in Toronto and two institutions in Tokyo. So university institution and affiliated psychiatric hospitals. And then if I can replicate these findings, so I'm relieved.
Just one, because it's lower in ventral, the other one's higher. Is
Yeah,
that mean
so-
opposite finding or
region
specific?
In terms of neuromelanin, for now we cannot replicate finding. But
It's different region.
so yeah, but I need to consider why this difference happened between our studies. And then we found, yeah, as you said, this study found dorsal substance Niagara. And this study found the difference in the ventral substance Niagara. And these two regions involve a bit different pathways in the dopamine pathway. So this is my current interpretations of the different findings.
Thank you.
All right, our next speaker is Professor Uite Kim from SNU. He's going to give a presentation titled Neuroimaging Fingerprint for Treatment-Resistant Schizophrenia.
Thank you. Good morning, everyone. I'm Uite Kim from Seoul National University. I'm very happy to make a presentation here today, and I'm very happy to see my friend Dr. Nakajima here. Many congratulations on the opening of SNUBIC Center today. Dr. Nakajima already made a comprehensive presentation about schizophrenia. So I would like to make a short presentation introducing how MRI can help understand mental illness, especially focusing on treatment-resistant schizophrenia. Yes. Let me further introduce myself. I'm seeing patients with psychotic disorder in my first clinic in Seoul National University Bundang Hospital. First means first episode psychosis clinic, and it is also the abbreviation of first individualized approach for safe treatment. Thus, first indicate both the subject and the purpose of this clinic. And with the patient in the clinic, I'm doing a pharmacoimaging study about mental illness in MPIC, which is my laboratory. Then what is pharmacoimaging? Historically, psychiatrists studied the pathophysiology of mental illness based on the symptoms like visual hallucination, auditory hallucination, obsession, and compulsion, and so on. Meanwhile, a drug was found to be effective for treating the symptoms of mental illness, which had been difficult to be managed. So scientists studied the mechanism of action of the psychotropic drugs and proposed hypothesis explaining the pathophysiology of mental illness based on the mechanism of action. Dopamine hypothesis in schizophrenia, serotonin hypothesis in OCD, and GABA hypothesis in mood disorder can be exampled. So drugs used for treating mental illness can be a window for understanding biology of mental illness. Nowadays, we have neuroimaging techniques which enable us to observe the brain in the living human. So pharmacoimaging is a field of measuring psychotropic drug effect on the brain by using imaging technique. Schizophrenia shows many symptoms like thought problem, perceptual disturbance, like auditory and visual hallucination, cognitive dysfunction, and even movement problem as seen in catatonia. Though schizophrenia is currently a well-documented and understood brain disorder, it was not recognized as such until relatively recently. Until recently, patients with schizophrenia were considered to be possessed by a spirit or devil. So they were secluded and imprisoned in an asylum and undressed, chained, and tortured. Meanwhile, a breakthrough was made by Dr. Henri Laborie. This is the picture of Dr. Henri Laborie, who was a French neurosurgeon. He used chlorpromazine for tranquilizing patients for surgery. From his clinical observations, he discovered chlorpromazine is also effective in calming patients with schizophrenia. It was a kind of a chance discovery. Though he discovered the antipsychotic effect for the first time in chlorpromazine, the mechanism of action was not known at the time. After the discovery of chlorpromazine, many antipsychotic drugs were developed, structurally similar to chlorpromazine. But still, the mechanism of action of all the antipsychotic drugs was obscured. This study is about the relationship between the binding affinity of antipsychotic drugs to dopamine receptors and their average clinical dose. As you can see here, there is a beautiful linear relationship. And from this result, dopamine hypothesis was proposed, and the mechanism of action of antipsychotic drugs was thought to be involved in dopamine system. This could be an example that pharmacological treatment informs on the understanding of biology of schizophrenia. The study shown in the previous slide was conducted in vitro. At the time, it was not possible to measure something like dopamine system in the living human brain, but the introduction of neuroimaging techniques enable us to measure change by antipsychotic drugs in the living human brain. This left picture is showing the regional brain activity measured by using FDG PET in the living human brain. And this graph is the comparison of frontal lobe activity between healthy control, non-schizophrenic psychiatric patient and the psychiatric patient. As you can see here on the slide, patient with schizophrenia showed reduced activity in frontal lobe. It is called as hypofrontality in schizophrenia. Additionally, on the right picture, regional specificity of dopamine receptor distribution across the brain was found. There are 5 types of dopamine receptors, and dopamine D2 receptors are found to be rich in the striatum, as you can see here. And the D1 receptors are rich in frontal lobe, as seen in the right graph. The hypofrontality in schizophrenia and this kind of study help shape the dopamine hypothesis by adding the regional specificity into the defective dopamine system in schizophrenia. Specifically, the positive symptoms like delusion and hallucination is thought to be related with hyperdopaminergic activity in major limbic pathway. And in the case of negative and cognitive symptoms, the major cortical pathway dysfunction is considered to be causally related. In vitro and in vivo studies have informed us about the abnormality of dopamine system acting in pathophysiology of schizophrenia. In this context, we need to have a look at the mechanism of action of antipsychotic drugs. As mentioned earlier, all the antipsychotic drugs are binding to dopamine D2 receptors, which are located in the postsynaptic area. And the antipsychotic effect by antipsychotic drugs in schizophrenia can be explained in 2 ways. The first one is that pathophysiology of schizophrenia is related with higher expression of dopamine receptors in the postsynaptic area, and that the antipsychotic drugs block the receptors, reducing the number of active receptors. The other one is that pathophysiology of schizophrenia is related with presynaptic dopamine synthesis or release, and that the antipsychotic drugs reduce dopaminergic neurotransmission from presynaptic area to postsynaptic area by blocking receptors. The consequence observed as antipsychotic effect would not be different, but it is important to see which one is the primary locus of dopaminergic abnormality in schizophrenia. So far, the postsynaptic receptors have drawn attention as a pathophysiology of schizophrenia, since antipsychotic drugs are dopamine D2 blockers. Indeed, some researchers reported a higher density of D2 receptors in schizophrenia. However, it was attributed to the exposure to antipsychotic drugs. Antipsychotic drugs induced upregulation of D2 receptors in postsynaptic area. This meta-analysis is pointing to the presynaptic area as a primary locus of dopaminergic abnormality. Meta-analysis revealed high presynaptic dopamine synthesis or release in schizophrenia. In summary, the pharmacoimaging technique has advanced our understanding of schizophrenia as from possession by spirit to presynaptic dopaminergic abnormality at the level of a synapse, shaping the hypothesis clear. The presynaptic dopamine activity may predict psychotic relapse in remitted patients with schizophrenia, which is showing the importance of dopamine activity in understanding and treating schizophrenia. This graph is showing the presynaptic dopamine activity in remitted patient with schizophrenia after antipsychotic discontinuation. As the red line is patient who relapsed after antipsychotic discontinuation, and green line is patient who showed no relapse, and the black line is healthy control as a control sample. And as you can see here, the patient who relapsed after antipsychotic discontinuation showed increased presynaptic dopamine activity after antipsychotic discontinuation. Considering the presynaptic dopamine abnormalities underlying psychotic symptoms, it is plausible that pharmacological action of antipsychotic drugs may be exerted by blocking postsynaptic dopamine receptors. And consequently, reducing the overflow of dopaminergic neurotransmission from the presynaptic area. If increased presynaptic dopamine activity can explain all the pathophysiology of schizophrenia, all the patients with schizophrenia should be ameliorated by the current antipsychotic treatment. However, we are seeing patients refractory to current antipsychotic drugs. It is a treatment-resistant schizophrenia, and the treatment-resistant schizophrenia is defined as patient with inadequate response to adequate treatment trials with at least 2 different first-line antipsychotic drugs. It is reported that the prevalence of treatment-resistant schizophrenia is between 15 to 30%. Schizophrenia can be divided like this based on antipsychotic responsiveness. Treatment-responsive schizophrenia, treatment-resistant schizophrenia, TRS, and ultra-treatment-resistant schizophrenia, who are not responsive to even close-up in treatment. It is hard to differentiate these groups by clinical manifestation or symptoms. This difficulty causes delayed treatment and poor prognosis in treatment-resistant schizophrenia. We have the only antipsychotic drugs for treatment-resistant schizophrenia. It is clozapine. As I mentioned, it is not possible to disentangle treatment-resistant schizophrenia from treatment-responsive schizophrenia based on clinical manifestation. So, all the patients with schizophrenia are treated with first-line antipsychotics. And then they were diagnosed as having treatment-resistant schizophrenia based on the antipsychotic responsiveness. It's a kind of trial and error strategy. This kind of strategy causes treatment delay in initiating clozapine treatment and risk our patients with unnecessary high doses of first-line antipsychotic drugs. Then, what would be the dopamine activity like in TRS? As I mentioned, the reason why schizophrenia can be treated with D2 blockers is that schizophrenia involved high presynaptic dopamine synthesis or release. Then we can imagine TRS is not likely to be related to high presynaptic dopamine activity, while their clinical manifestation is similar to treatment-responsive schizophrenia. Indeed, studies, you already saw this slide in the previous talk by Dr. Nakajima. Studies reported that treatment-resistant schizophrenia did not show as high presynaptic dopamine synthesis as treatment-responsive schizophrenia did. And the structural and functional connectivity regarding presynaptic dopamine synthesis was different between treatment-resistant schizophrenia and treatment-responsive schizophrenia. Dr. Nakajima reported a systematic review about neuroimaging finding in treatment-resistant schizophrenia, though he said there is the consistent finding are lacking in treatment-resistant schizophrenia. He reported hypometabolism in prefrontal cortex and hypometabolism in basal ganglia. Please, these two characteristics of TRS in mind, because I will mention these two characteristics later. And Dr. Nakajima group also reported glutamatergic and GABAergic metabolite level according to the antipsychotic responsiveness. It's a bit complicated, but Dr. Nakajima explained these results in the previous talk. And as mentioned earlier, the only antipsychotic drug which can be used for treating treatment-resistant schizophrenia is clozapine. In this context, clozapine can help us understand the pathophysiology of treatment-resistant schizophrenia. The pharmacological characteristics of clozapine are distinct from those of the rest antipsychotic drugs. As you can see here, clozapine shows a very low dopamine D2 receptor occupancy and a strong antagonism for D4 receptors. Adding to the previous slide, clozapine seems to interact with a glutamate system as well. This is the brain of rat, and ketamine altered the brain. Ketamine altered the brain, turning the color black. However, clozapine prevented the ketamine from turning the color black, while haloperidol could not. Okay. As you may already know, ketamine interacts with glutamate system. So the effects of clozapine on ketamine-induced alteration suggest that clozapine is acting on the glutamate system as well as dopamine system. Indeed, in vitro studies support this. NMDA receptors are regulated in vivo by glycine, which is regulated in turn by glycine transporters. This is showing the relationship between clozapine level and glycine uptake. This is the concentration observed in plasma and in the brain here when treating with clozapine. As you can see here, clozapine inhibited glycine transporter in a dose-dependent manner. Taken together with the result in the previous slide, this is indicating clozapine may exert antipsychotic effect by acting on the glutamate system, and this may contribute to the differential efficacy, clinical efficacy of clozapine compared with other typical or atypical antipsychotics. And the treatment-resistant schizophrenia is likely to be related to glutamatosic abnormality. So, to examine the clozapine effect on the brain and to reveal the pathophysiology of treatment-resistant schizophrenia, researchers, including me and Dr. Nakajima, organized a consortium called as CLIMATE. It is clozapine imaging and treatment effects. So from now on, I will show you some results from CLIMATE. We measured the clozapine effect on the brain by using MRI. So as you already know, MRI is sensitive to macrostructure change in the brain. So the first one is cortical gyrification. Cortical gyrification or brain folding is a morphological process in which the cerebral cortex folds during the development. When examining the trajectory of gyrification development in healthy individuals, it begins during the fetal development, with most of cortical folding occurring around the prenatal and perinatal period, after which it remained relatively stable throughout the life. Therefore, changes in gyrification reflect cortical abnormalities arising from early neurodevelopmental processes and can serve as a potential biomarker of early neurodevelopmental risk factors. This study was analyzed by Dr. In-Kyung Park, and in this study, the local cortical gyrification was measured using vertex-wise analysis and compared between patients with treatment-resistant schizophrenia and first-line antipsychotic responder at baseline and after 18 weeks of clozapine treatment. The baseline analysis here showed significantly reduced gyrification in the left and right precuneus regions and right inferior frontal regions in treatment-resistant schizophrenia compared to first-line antipsychotic responder. And the follow-up analysis revealed that the reduction of gyrification index in the same region remained unchanged even after clozapine treatment, suggesting it is unaffected by antipsychotic medication. These findings suggest that reduced gyrification in this region may serve as neurodevelopmental deficit marker specific to treatment-resistant schizophrenia. And we examined their correlation with the reasoning and problem-solving score. The reasoning problem-solving score is known to be closely associated with neurodevelopmental dysfunction. So this analysis aimed to provide additional support for the idea that reduced gyrification observed in TRS, treatment-resistant schizophrenia, represent neurodevelopmental marker. At both, and this is the baseline comparison and the follow comparison. Treatment-resistant schizophrenia showed a significantly lower score of reasoning and problem solving compared to first-line antipsychotic responder, as you can see here. Moreover, significant positive correlation was found between it's too small gyrification and reasoning and problem-solving performance. Meanwhile, we also look at the different structural change with T1 MRI. While the cortical gyrification remained unchanged or stable during clozapine treatment, the gray matter volume and cortical thickness exhibited substantial change over time. This contrast suggests that cortical thickness may be more influenced by external or environmental factors, whereas gyrification appears to be less affected by medication and may instead reflect more neurodevelopmental and genetic characteristics. And it is plausible that the macrostructural change would be preceded by microstructural change in the brain. In the living human brain, we can examine microstructural change induced by clozapine using MRI texture analysis. Actually, texture analysis was analyzed the patterns of gray signal intensity, which are potentially associated with cytoarchitecture and neuronal density. Texture analysis was originally used in computational vision, but now texture analysis has been applied to many medical fields like cancer diagnosis. Texture analysis quantifies the relationship of neighboring voxel intensity. So it quantifies the quality of signal intensity, the neighboring Box cells. This slide is showing the change of GLCM correlation extracted from the texture analysis in the caudate nucleus. The left graph is regarding the change in the caudate nucleus, which is one of key regions associated with schizophrenia. The green and red line are treatment-resistant schizophrenia. And they were treated with clozapine. And the black one is first-line antipsychotic responders as a positive control. And as you can see here, clozapine induced reduction of this parameter. Here, However, at baseline, the correlation indexes were different according to the clozapine responsiveness in treatment-resistant schizophrenia. The green line is treatment-resistant schizophrenia who were responsive to clozapine treatment. The red line is treatment-resistant schizophrenia, but who were not responsive to clozapine treatment, called as ultra treatment-resistant schizophrenia. As you can see here, at baseline, the index was different between two groups. So this graph is reflecting the clozapine's effect on the microstructure in the brain. And it is also showing the heterogeneity of treatment-resistant schizophrenia in the aspect of pathophysiology. The previous result is further supported by diffusion kurtosis imaging study. DKI technique builds on DTI. Instead of assuming that water diffusion follows a simple Gaussian distribution, DKI measures how much the diffusion deviates from that Gaussian distribution, what we call kurtosis. So why does that matter? Because this non-Gaussian diffusion behavior turns out to be sensitive to complexity and heterogeneity of tissue microstructure. Our DKI study in treatment-resistant schizophrenia revealed that the diffusion kurtosis between orbitofrontal cortex and thalamus was reduced following clozapine treatment, and the mean kurtosis value were related with symptomatic improvement. This data was primarily analyzed by Dr. Moon Sun-young here. And then what causes such microstructural changes after initiating clozapine treatment? There might be many factors influencing the microstructural change by clozapine. They include dendritic remodeling and neuroinflammation like this. Indeed, there have been reports about increased inflammatory markers in the blood in patients with schizophrenia, and the modulatory effect of clozapine on the neuroinflammation has been also reported. However, it's a kind of still speculation. It still remains to be confirmed in the future study. We also examined the functional activity change by clozapine. in treatment-resistant schizophrenia group. We measured regional brain activity using resting state fMRI and analyzing a rehole. We found the difference in functional change across the brain regions, namely the subcortex, including the striatum, exhibited decreased activity like this after the treatment with clozapine, while cortical area showed increased activity. In the previous slide, I mentioned two characteristics of treatment-resistant schizophrenia. One is hypometabolism in frontal lobe, and the other one is hypermetabolism of in the striatum. Considering the two characteristics, the clozapine here induced changes in the opposite way. So it can be said that clozapine may normalize the pathophysiology of treatment-resistant schizophrenia, as you can see here. The first-line antipsychotic responders who did not expose to clozapine did not demonstrate such changes, as you can see here in bar graph. This is clozapine group, and this is the first-line antipsychotic responder group. This data was analyzed by my PhD student, Haejin Shim, here. She is attending here. And in patients treated with clozapine, we speculate that unlike typical D2 antagonist, clozapine simultaneously enhances cortical activity and mitigates corticostriatal imbalance, representing a bi-directional neuroadaptive effect. It can be a unique mechanism that may underlie its clozapine's efficacy in treatment of treatment-resistant schizophrenia. Indeed, the functional change observed in the previous slide was associated with the psychotic and cognitive symptom improvement in treatment-resistant schizophrenia. Further, the cognitive improvement was specifically observed in clozapine responsive schizophrenia, while so-called ultra treatment resistant schizophrenia still remained unchanged even after the initiation of clozapine treatment. This graph, the green is treatment clozapine responsive schizophrenia, and this is the ultra treatment resistant schizophrenia, and this graph is showing the change in the aspect of cognitive function. And as you can see here, the treatment clozapine responsive schizophrenia showed the improvement after the initiation clozapine treatment, while the ultra treatment schizophrenia, treatment-resistant schizophrenia didn't show that kind of improvement. So in summary, dopamine hypothesis was developed from the mechanism of action of D2 antipsychotic drugs. But there is so-called treatment-resistant schizophrenia, TRS, who are not responsive to first-line antipsychotic medication. Treatment-resistant schizophrenia need to be treated with clozapine. The treatment-resistant schizophrenia shows a different brain macrostructural and microstructural change by clozapine. The change of microstructure and functional activity induced by clozapine can address the pathophysiology of treatment-resistant schizophrenia. Yes, thank you for your attention, and this is my presentation. That's it. Thank you.
Thanks for the great talk, Professor Kim. We have some time for questions. Please raise your hand and ask any questions. Question over there.
Thank you for the interesting talk. My question is that I might have missed this, but what is your hypothesis on the ultra treatment responsive schizophrenia, unlike because it couldn't be treated by clozapine?
Yeah, it is a very important question, and we need to address that question in the future study. But it is speculated that GABA system can be involved in the ultra treatment resistant schizophrenia, while most of treatment resistant schizophrenia is involved in glutamatergic system, but it is not yet investigated. So we need to conduct such kind of study in the future. Okay.
I see. Thank you.
Thank you for great lecture. I'm not familiar with schizophrenia, but after listening your talk, I can guess that the dopamine regulation is very important, and the dopamine hypothesis is based on the treatment methods. Actually, my question is that after treatment of the dopaminergic regulator, I mean the schizophrenia medication, I can guess that there is the changes, maybe pathological changes in the dopaminergic pathways, like the Parkinson disease. So is there any changes in striatum or substantia nigra after treatment?
Yes. It is a very important question because, as I mentioned, all the antipsychotic drugs are acting on the postsynaptic dopamine receptor, and the primary locus of dopaminergic abnormality is pointing to presynaptic dopamine activity. So it is whether the current antipsychotic drugs can normalize presynaptic dopamine abnormality by blocking postsynaptic receptor is very important issue, because it can be upregulate of presynaptic dopamine activity, while the psychotic symptoms are ameliorated. But there are inconsistent results about that. Some researchers reported increased presynaptic dopamine activity after the initiation of antipsychotic treatment. but German group reported the down-regulated presynaptic dopamine activity with the antipsychotic treatment. There is no consistent result, and which should be addressed in the future study.
Any more questions?
Thank you for your talk. I was just wondering to what extent, even in the patients that are treatment responsive or the clozapine responders in the treatment resistant, to what extent, when you talk about efficacy or responder, how much restoration of function is there, and how much is still not addressed in relation to kind of symptoms or general quality of life of the patients. So in other words, how much further, even in the treatment responsive patients, is there room for improvement of the treatment methods and the current first line treatments?
How much?
Room for improvement, so even if you're seeing responsiveness, obviously, presumably the restoration of function is not complete. So I'm just wondering, what is the gap between current treatment efficacy and where, one needs to be ultimately, if that makes sense.
Sorry, are you asking about the clozapine efficacy? Or?
I'm asking about in general. So, even in the treatment responders, right? So you would define that they're responding to treatment because there's an improvement in their symptoms. But presumably that improvement is not restoring, 100%, right? That they're not. So I'm trying to understand what the gap is between the current, state-of-the-art treatment, whether it's in the clozapine, in the first-line non-responders, or the ones who respond to classical antipsychotics. Just wondering, what is the gap with the current state-of-the-art, and where one would want to be ultimately, even in those who respond to treatment?
Yes. It is hard to define the target of improvement. We in the current study, we compared the treatment efficacy based on the symptom before the treatment. So clozapine efficacy is also compared, defined in comparison with the baseline symptom level before the initiation of clozapine treatment. Okay?
Yeah.
So,
oh, please.
They know. So, and functional recovery is mainly related to negative and cognitive impairment. And currently, so antipsychotics, as Professor Kim said, so antipsychotics mainly focus, target on positive symptoms, including delusions and hallucinations. So that's why, presentation, Professor Kim or I conducted is mainly focusing on positive symptoms. And first episode, patient with first episode schizophrenia, respond very well to antipsychotic treatment. So remission. So remission rate is very high, 80%. But during the follow-up, for example, 25 years, still recovery rate, especially consistent recovery rate is, transient recovery rate is maybe 25%. But continuous recovery rate is much lower, maybe 10%. social recovery means social functioning
Yeah.
or occupation or
Yeah.
marriage.
Yeah, yeah, right.
Is it the answer
Yes,
you
that's
require?
that's what I'm interested.
Okay.
So there's clearly a lot of space for,
Yes. Yeah, that's why we need
yeah.
to continue to
right,
continue
right.
research.
Yeah, I see. Yeah, yeah, yeah.
That's great. Do we have any questions coming from online?
I have one more question here.
Dr. Zhang?
Just probably quite trivial thing in terms of you got these 5 different dopamine receptors, say D1, say D2, you mentioned that. Is D2 the same for different people?
Sorry?
Is D2 receptor basically essentially the same between different people? Is that true? Is that the reason I'm asking is I wonder whether the lack of responsiveness may be to do with some individual differences in their receptors, so their binding of the drug.
You mean
if
the-
efficacy is different, for example.
You mean the D2 receptor?
For example, yeah. So a clozapine might bind to them at a different efficiency, depends on who you are. In a sense, there are some small but sufficient variation in the structure that leads to the biomechanical function is a bit different. Has that something being explored?
I'm not sure about it. Yeah, there. I have not seen that kind of report, but it is plausible, I think.
And I kind of
Because
really
it
go
is-
down to my real question really is, do we know the efficiency, for example, the delivery of the drug in the brain,
Mhm.
in the midst of the neurons, and then their binding efficiency? Do we have a good way to actually quantify that on an individual basis?
Actually, you are mentioning the pharmacokinetic profile of psychotropic drug. So yes, there have been reported about the inter-individual variation in the aspect of a pharmacokinetic profile of psychotropic drug, and it is that's why we need to individualize the treatment in the aspect of dose and the kind of antipsychotic drugs. So, but it is still based on the trial and error strategy. There is no biomarker to predict antipsychotic effect at the level of individual.
I understand that. What I meant, whether there's a way to determine the non-responder, is it because of those clozapine wasn't delivered enough into the brain, or wasn't binding to the dopamine receptor in those individuals as well? Or, is that something that can be actually quantified or directly measured?
Yes, there are some study about the occupancy, receptor occupancy by clozapine, but it is hard to conduct the occupancy study in every clinical trial. We assume the pharmacokinetic profile is similar across the patient, because based on the plasma concentration of measured in the
Blood.
yeah.
But not in the CSF. Right. Okay. Thank you. Yeah.
Okay, it's almost 11 o'clock. Do we have any questions from online?
No.
No. I have a quick question. So it was very interesting to see that the less cortical gyrification is associated with schizophrenia. Is that different etiology, or does that have to do with dysfunction in the dopaminergic system?
I didn't show the relationship between gyrification and genotype. We found some significant relationship between specific genotype and lower gyrification index. which indicate lower gyrification index is related with genetic difference. Affecting, yeah. So though the treatment-resistant schizophrenia looks similar in the aspect of clinical manifestation, the pathophysiology or etiology could be different from the treatment-responsive schizophrenia. The genetic, the specific gene was related to the dopamine system. So that's why we are the treatment-resistant schizophrenia is related with neurodevelopmental deficit.
Okay, that's it for the morning session. We're going to have lunch hour until 1:30. All right. How's everybody doing? Good. Okay, it looks like we got more audience this afternoon. Let's go ahead and get started the afternoon session. Okay, so our first speaker for the afternoon session is Professor John O'Doherty from Caltech, United States. He's going to give a talk titled Hierarchical Computations Underlying the Construction of Value.
Thank you very much. And I'd like to thank the organizers and my collaborator, Professor Anne, for inviting me. It's great to be here. It was wonderful to see the new scanner and wonderful to see that the technology for imaging is advancing and we can go on to new and exciting new directions. What I want to talk about today is there's been a long history actually for the last 20 years in the field of decision neuroscience or neuroeconomics, in which we've tried to understand how is it that people make choices between very simple things, right? How do we make decisions? So for example, between this apple, this orange, and this hamburger, what's going on in the brain when we make these kinds of choices? And for more than 100 years, economists have suggested that what's going on is that you're kind of maximizing some expected utility. So, you know, you're looking at the expected net benefit and the net cost and trying to choose the option that maximizes your overall expected net gain, right? Net utility. And what's kind of been quite remarkable over the last 20 years is that while economists, many economists, or if not most of them, consider this just to be an as-if thing. So it's just a kind of convention, a mathematical convention that you're kind of maximizing some function, which they're going to call utility, but they don't have really any expectation as a real thing called utility, or I'm going to call it value in the brain. But actually it turns out that if you scan people's brains while they're doing a task like this, making simple choices, you can see signals that kind of scale according to how much you want or like or value the goods. Okay, so for example, here maybe I want the hamburger more, right? I value that more. And there's some kind of representation in the brain that's scaling like this. And we've been able to test this with, this is an example experiment from my lab, but there's many, many, many experiments like this. And in this particular experiment, we gave people the option of purchasing a food item or a non-comestible good, such as a DVD or a t-shirt. You can tell this is old now. Who buys DVDs anymore? Or, you know, access to playing a lottery to win a monetary gamble. And we were able to find that there's bits of the brain when we use a measure of people's value, which we obtain behaviorally. There's different ways to do that. I'm not going to go into it. What we can find is that there's activity scaling with value for different kinds of goods. And we see this in most robustly in the ventromedial prefrontal cortex, okay? And in fact, you can look at the overlap between the activations for these different kinds of goods. And you see a kind of a common area of VMPFC that's really just correlating with value. And it's very robust, and there's many meta-analyses that kind of have kind of shown how consistent this is across studies. Okay, but one of the big questions that I've been interested in over, I guess, my lab members over the last 5 years or more is how are decision values actually constructed in the first place, right? How do we form these values in the brain for things? And, you know, the way we think about how we process stimuli, you know, when it comes to sensory, in the sensory milieu, we think about stimuli having attributes, right? So, for example, this triangle has color, has shape, right? It has size, right? So you can imagine there's all different kinds of attributes of a stimulus, and we think of, you know, our visual system, for example, as processing different elementary components, elementary features or attributes, and then across the cortical hierarchy, building up representations of more kind of complex objects, right? And so one question is, you know, does the brain do the same thing when it comes to assigning value to things? So, for example, this cake, maybe it has carbohydrates, it has fats, it has proteins, it has vitamins, it has different underlying features, right? And maybe we assign value to things by integrating over those features. So, for example, if we take these particular attributes here, maybe we assign weights over them. And in a simplest possible model, you could just linearly integrate over the weights for each level of each feature and then compute an overall value, okay? So what's going on here is it's kind of an active construction of value for things. And, you know, we do also talk about learning value, you know, for example, using reinforcement learning through trial and error. And that's certainly going on, of course, as well. But the idea of actively constructing value is quite useful because if things change, right? So, for example, imagine I'm valuing the cake when I'm really hungry, okay? Middle afternoon, you've listened to me for too long. It's a while since lunch, so maybe the cakes and desserts outside sound really appealing, or cookies. But supposing you've just had lunch, you've had a massive dessert of cupcakes, okay? Well, then your value for this cake is going to be lower, right? It's going to be changing. So our value can change flexibly depending on the context we're in or our internal motivational state. And maybe there's a way of flexibly changing either the weights, right, which is what you can see here, modulating those over, say, for example, carbs and fats, which might be what these kind of foods contain. Or we could actually actively modulate the value of carb or the value of fat directly, and that would change flexibly our overall value for other things, okay? And so one of the questions that we were first interested in is, well, could we understand just exactly this problem? How do we construct value for things like food rewards? So this was a study that Shinsuke Suzuki and Logan Cross did in my lab actually a good while ago now. And what we did was we presented different foods, about 56 different food items. And we had people, we gave them an endowment, a cash endowment, and we said, well, how much are you willing to pay for this particular food, okay? And one of the foods we would take at random, and they get to consume that at the end of the experiment. So they come into the experiment kind of hungry, motivated for food, and we track their value computation. So very similar to the study I showed you just earlier. But the other thing we did was we got people's beliefs. So after the experiment was over, we probed people to tell us, well, how much fat do they believe is contained in the stimulus, how much salt, how much carbohydrate, and so on, protein, vitamin, and so on. So we've got a whole bunch of different attributes, and they're not even all listed here. And we then used those attributes to see, could we predict people's values from when they were in the scanner, okay? So that they didn't know we were asking them about the attributes of the food when we just asked them to make the value judgment. So we're just going to see, can we predict, based on their ratings, their beliefs over value? And it turns out we can. So we can do pretty well, like the correlation between the actual value that they give in terms of the monetary amount and an integration over their attributes, okay? A model that's integrating over their attributes. What we're doing is integrating over a subset of attributes that are best predicting the value. And we'll actually do pretty well. If we take their subjective beliefs over the attributes, it does better than if we just take the objective nutritive attributes, which are, you know, what you might get from the FDA label on the food, for example. And it's not just driven by overall caloric content, okay? So it seems like we could predict behavior from the attributes, and then what's going on inside the brain? Well, what we did is we, while they're making the judgments of value over the food, and they're not in the scanner making the judgments of the attributes, okay, importantly. And they don't know we're going to ask about attributes. But we then said, well, can we decode so we can train a classifier, okay, to detect multivariate patterns that distinguish between high versus low protein content, high versus low fat content, and so on. What we're showing you here is not for the nutritive attributes, but just for value. Can we predict high versus low value? And again, we can see, once again, the VMP of C here, that's correlating, that's having a representation, a multivariate representation in this case, of value. But we also see something more laterally as well. So value seems to be present in the orbitofrontal ventromedial prefrontal cortex. But then, can we decode the actual nutritive attributes? And it turns out we can for at least 4 of those attributes: fat, carbohydrate, protein, and vitamin content. We can decode that from the lateral orbitofrontal cortex. So you can sort of see here just decoding from the overall region of interest, defined over the whole lateral OFC. And here it's kind of using a searchlight. We're moving kind of a moving searchlight and decoding from there. And we're just kind of looking at the plotting these average accuracies across participants. And you can see, again, we can decode these different attributes, but not from the medial OFC, just from the lateral OFC. Interesting. Okay. Now, one question could be, well, does this represent the kind of visual content of the food? Okay. The kind of the visual features that the food is composed of. And maybe one way we were thinking about this at the time was, well, this is a confound. The kind of visual features might be confounding the nutritive attributes. And so we basically also extracted various image statistics from each of the food images. And we tried to see, well, could we decode those? Things like luminance, hue, saturation, brightness, and so on. And we could decode that from area V1, primary visual cortex, but not from the lateral OFC. And that was sort of giving us the suggestion that the lateral OFC representation really is more of a higher order feature of the nutritive content of the food that's being used at the time of decision making. And the other thing we could do is to see, well, if we take the nutritive features and we look at the time of decision making, changes in connectivity between, say, lateral OFC and the feature representations and medial OFC and the feature representations. We could see, well, is there an integrative process by which feature representations are being, the coupling between those and these value areas is changing as a function of the time of decision making. And what we found was that we could pretty much across all the features, fat, protein, carb, vitamin, those representations, they were increased coupling with the value signal in medial OFC at the time of decision making, much weaker effects between the value signal in lateral OFC and the nutritive features. So that led us to kind of conclude, or hypothesize at least, that maybe what's going on is that there's these nutritive features in lateral OFC that are being integrated over to compute an overall value in the medial OFC. Okay. So from there, we decided to get even more complicated. And this was a project that Kyo Igaya in the lab, who joined the lab after Shinsuke had left to go on to make his own faculty position. And Kyo was interested in kind of amping up the complexity to looking at something like art, okay, or images in general, okay? So this is obviously a very high-dimensional complex stimulus. And the question is, how do we form value judgments for something like this? And can we build a model that allows us to predict people's value for a piece of art that they haven't seen before, or that the model hasn't seen before? And so we came up with the same kind of scheme, which is that there's going to be some transformation of the input that will allow the brain to compute value. And then the question is, what's the nature of that transformation that's going on? And we were inspired by the food study. We said, well, maybe what's going on is that there are different attributes or features being represented, and that the brain is going to combine over those features. And, you know, the simplest model, though it's not the one I think is really going on, but the simplest model would be a linear integration. And we thought about there being different, just like there was for food, different levels of features, different kind of complexity of features. You can think about low-level features as basically being those kind of very elementary visual properties, okay? And you can extract those using a computer vision algorithm, and it'll extract a whole bunch of different things. So here, over 71 different features. We also extracted our own additional features beyond the one that the model was giving us. So the examples are things like mean hue, mean saturation, mean lightness. You can also have segmentations of the image, and you can have, again, those features over the individual segments and so on. And then you can also imagine that there's more high-level features which are relevant for aesthetic judgments. So that could include, you know, is it concrete or abstract, right? The art. Okay, is it dynamic or still? Does it have a hot or cold temperature? Okay, color temperature. Is it good or bad, right? So does it have a positive or negative affect? So the affect in the image doesn't correspond to the same thing as the affect that you feel when you see it. So for example, if you go and see Edvard Munch in Oslo, you know, the Scream, the famous painting of his, that's obviously conveying a very negative affect, but you might feel a positive affect looking at that, right? So those are kind of what we call kind of high-level features, and we could extract those, get people's beliefs over those features, judgments over those features. So we had a separate set of participants judge a set of art images. We actually had about 1,000 images, art images, and we had people kind of judge these high-level traits based on that. They actually had some art experience. They were students training in art, okay, from the local art school, in Pasadena. And then we had people do a study like this, where they just saw different paintings. We had over 1,000 different paintings, as I said. These came from Wiki Art, and we also had some other paintings that were included from another set of images that another group had worked with. And they just make simple ratings, 0 to 3, while they're in the scanner, okay? So some people were scanned. Well, actually, this is just behavioral study. So they're just actually sitting at a console, seeing the images and making judgments. We also ran some studies online as well with a whole bunch of additional participants. So this is just purely behavior. And here are 7 participants who are in lab. This was not in the scanner, I should say. And we're saying, well, can we train a model? So we take a whole bunch of these features, okay? And we assign weights over the different features. We use a kind of a lasso regression. So we're coming up with a set of features that are best predicting the people's value. And obviously we do cross-validation. So we can either train within a participant on some portion of the data. It's a cross-fold validation. I think it was 5-fold. And then we test on another portion of the data. Or we can even just train the model on other participants and test on a given participant. Or we can even train the model on people who did the study online, okay, and then test the people that were doing the ratings in the lab. And what you can sort of see is that a linear model trained on integration of these feature weights is actually predicting people's value judgments for art images actually pretty well. So you can see there's a variation. So, you know, one person were doing very, very well indeed, and other people were doing less well. And there's actually an explanation for that, which if you're interested, I can talk more about it. But the point is that we're not obviously doing perfectly well, right? There's a lot of variance we're not explaining, but we're very significantly above chance explaining people's liking ratings based on just this integration of the features for a held out piece of art. And the same thing applies if you test them online, if you use online data, okay? I'm actually going to skip this for now. And I'm going to go into the brain. So the question then is, well, how does the brain construct value? So we basically put people in a scanner, and here we have 6 participants, and they're doing the same task. It's essentially the same task. And each person is rating over 1,000 images. And so what we're doing here is more of a deep fMRI study, where we're taking a small number of people and scanning them for a long time. So each person is scanned over 4 different days for an hour per day. So we're getting over 4 hours of data per person. And once again, we can train the model on their behavior while they're viewing the art images. And again, we can predict their ratings better than chance. So that's all good. And then we can look in the brain and see, well, are there value signals being computed that are correlating with the liking ratings for the art? And we'd, of course, expect to see this. That's been reported in many previous studies. And indeed, we do. We see, especially in medial parts of the frontal lobe, as ventromedial, also extending up a bit dorsally in some people, we can see value signals. So these are areas scaling with how much you like the art, okay, positively. What we can also see is, well, are these features, these individual features, also represented? And what we did was we broke down the features into the kind of low-level features, which were the kind of these very elementary visual statistics, and also these high-level features, which are these kind of things like, as I said, is it dynamic or still? Is it concrete or abstract? And what we're showing you here is we're just moving through an atlas for the character, basically identifying regions of interest in the visual system. So we're going kind of from V1. This is actually the ventral visual system on the left, and on the right is the dorsal visual system. And we're just basically looking at the number of voxels that are correlating either with some combination of high-level features or some combination of low-level features. And basically what we're doing is looking at the kind of the ratio of low to high-level features. So what you can see is that, which is what we'd expect, is that as you go from early visual cortex, these are each one of these plots is a separate subject, 1 to 6. Especially if you look at the ventral visual stream, basically the blue is the low-level features and orange is the high-level features. And you can basically see that we see greater representation of low-level features early in the visual system. And then as you move through to more higher-order visual areas, then you're getting more and more representation of the higher-level features, which is what we'd expect. Okay, so maybe that's not surprising. A little bit less clear, but still in the same trend in most people in the dorsal visual stream. And we can also look in places like frontal and parietal cortex. And what we were maybe expecting to see was that we would see mostly high-level features there. But actually, we're seeing kind of a mix of both high-level and low-level features being represented throughout parietal and frontal cortex. But if you look at value signals, this is objective value. And what we're doing here is plotting both high and low-level features here in kind of in green, and subjective value here in kind of red. And basically, as you go through from early visual to higher order visual, parietal cortex, lateral prefrontal, and medial prefrontal, across these separate subjects, you can see that there's a gradual increase of the representation of value. So the value is most robustly represented when you're getting into kind of medial parts of prefrontal cortex, which is not surprising given the results we've already shown you. But it's suggesting that there's kind of this hierarchical process whereby you start off representing mostly the low-level features, then you start to represent the high-level features, and there's a gradual transformation into a value signal as you get into prefrontal cortex. And we can also do connectivity analyses where, again, we seed in the value region at the time of decision-making, and we look for increased coupling with other parts of the brain at the time of decision-making, which we would expect would be relating to the integration process. So if there's regions of the brain that are encoding these feature representations, and then they're sending that information and integrating over it to create an overall value signal, would expect an increase in coupling between those areas and the region encoding value. And what we're seeing is that actually we do see that. So we don't see coupling with the visual cortex directly, but with parietal and lateral prefrontal cortex, as well as parts of medial prefrontal in some people, we're getting increased coupling at the time of value judgment, okay, when they're making a judgment about the liking rating. So it suggests, again, that there's this integration between these kind of lateral prefrontal region and as well as parietal cortex 2 at the time of decision making relating to this feature integration to compute value. Okay, so we've done some art. Now I'm going to go back to food, because where we left the food was that we were thinking about how these nutritive features are represented. And we thought that, well, there's an integration of those nutritive features to compute value. But actually, you know, for food value, we also have this rather complex process going on, presumably, where there's these visual features of the food, which are then being integrated over. And there's going to be some higher order features, such as nutrition, which we've already shown, that are going to be relevant for computing value. But another feature set that we thought could be interesting would be taste features. So, for example, how salty or sweet, or you know, or bitter is the food, for example. Okay, and there's going to be many, many more features in that. So we're not going to capture everything, but we just wanted to look to see, was there a more richer process going on, and can we characterize the order in which this is happening? Right. So simple model would be going from visual through to taste, through to nutrition, and ultimately through to value, okay? And can we test that? Well, I'm not going to show you imaging data for this particular thing because that's still in process. But what we did do is we were able to get a database of food images that other people had collected, Blakehurt et al. And what they've done is they've obtained ratings for value for these foods. Actually, these are average ratings. So an average across a population of people. But we also had ratings for things like nutrients that were obtained as part of this database. And we could also actually obtain ratings ourselves in another set of people for things like taste components. Interestingly, you can also ask ChatGPT to do this, and it does pretty well, actually. So you can also use ChatGPT to extract these kind of taste and nutrient features. So we have that, and then basically want to see as well, let's analyze both the very basic visual features, okay, just like we did for the aesthetics. This is the work by Saida Sadehi in the lab. And she also kind of was interested in colors. So she extracted color information, color of the food, she thought would be relevant. And then we have taste features, as well as a texture as well, fat texture, mouthfeel. And we have the nutritive features that we already talked about. So we can basically use all of that to see, well, can we predict average ratings? And we can look to see, well, first of all, can we predict taste or nutrients from the visual features? And it turns out we can. So we can train a model to do that, and it does pretty well. So we actually looked at a linear model, which is what we've been using up to now. But then you could also fit a nonlinear model, a random forest. And you can sort of see that it actually does better. It's actually predicting a little bit better, suggesting that indeed there's some nonlinearities as well, which is no surprise into how these features are being used to predict other features. You can go from visual features to predicting pretty decently all of these kind of nutritive and taste features. And then you can take either visual features, taste features, and nutritive features, and you can predict value. And turns out that the best predictors of value is taste and nutrients together. You don't get much more information if you include the visual features as extra. So basically all the information you're getting that predicts value is coming from taste and nutrients, okay? And the colors actually also do actually allow you to predict some aspects of the features, such as, for example, how bitter something is. And this is just purely based on behavioral ratings. We can fit a causal model to see, well, what role causally are all these different features playing? And basically it supports their idea that, you can go from vision to nutrients and taste, and then from nutrients and taste, you can go to value. What you actually have here are two other high-level features called healthiness and tastiness. So this is also coming from the neuroeconomics literature. My colleague Antonio Rangel and his collaborators identified healthiness as another feature which people use when they're making decisions, especially in LA. Okay, they care about, is this food going to be healthy or not? And also, how tasty is it going to be? And some integration of those allows you to predict overall value. Because I might like that cake, but know that it's going to be very bad for me. So maybe I'll actually rate that as less valuable than something that's healthy. Okay. And so it turns out, even just from behavior alone, we can see that, this nutritive and taste components are being integrated over, being computed from the visual features, and then giving rise to this healthiness and tastiness, which in turn is then giving rise to value. So that's where we are. And actually what we're planning to do is to collect data with more temporal resolution, maybe going beyond fMRI to something like MEG, to try and see if we can understand the dynamics, the temporal dynamics by which these feature representations are computed. And we're also actively collecting single unit electrophysiology. So we have patients who are undergoing, just like we saw yesterday, intracranial EEG and single unit recordings. They're undergoing neurosurgical treatment for epilepsy. So they have these depth electrodes implanted, and this is in collaboration with Uli Rutershauser, Cedars-Sinai. And they're basically looking at food images as well as art images, actually. And we're going to look at to see to what extent are these different features represented in different parts of the brain. We're getting recordings from amygdala, hippocampus, posterior temporal lobe cortex, prefrontal VMPFC, cingulate cortex, pre-SMA, whole bunch of areas. And actually, I didn't present this data here because this is a neuroimaging symposium, but we see a lot of interesting representations in the amygdala of these taste features and nutritive features. It seems to really, really care. And that might be another region, along with lateral orbital frontal cortex, that's involved in representing the features, which are then being combined over to compute overall value. But that's still work in progress. Okay, so I'm going to conclude with this last study. Okay, so you remember that when I introduced this concept, I was talking about the active construction of value being useful because you can compute different values for things for the same stimulus, depending on maybe your internal motivation, okay? And another kind of thing is that might affect your value for something is the context in which you're using it, or the goal that you have for the item. So imagine this jacket, okay, this winter jacket. And imagine you're purchasing this online or considering purchasing it. You're assigning a value to it. But there's two different contexts in which you might be evaluating this thing. So one context would be to go on a ski trip, okay? Probably useful to have a warm coat on a ski trip, so that'll probably have a high value to you, okay? But instead, imagine that you're going on a beach vacation to Hawaii, okay? Probably not that useful to bring that warm jacket with you, right? Okay. So what the same stimulus is going to have very different value depending on the context of the goal, the overall goal that you have for your behavior. And so Anik Franzen and Kyo designed a task to allow us to look at this question. And so what we're doing is we had a whole database of clothing images, right? So these are kind of fashionable items or unfashionable, depending on your judgment. And obviously, I'm a big fashion person, as you can tell from looking at me. And so that's why I was very excited by this project. So you have people looking at these different clothing items. We had a separate set of clothing depending on whether you were identified as male or female, gender. And the task was that you see these items, these clothing items, and you basically have to say, well, how much would you like to purchase this item? You have to kind of give a rating. There's different contexts in which this is happening. So one is kind of a tropical island, okay? So for example, maybe the swimsuit would be a good thing for a tropical island, or sometimes a ski trip or a job interview, okay? So you're kind of purchasing them, imagining that you're purchasing them for these different situations. And then you give a rating of how much you would like to purchase this item, how much you like this item, okay? And so we have these different contexts. And what we also did was we came up with a bunch of features which we thought were relevant, okay? So warmth, the warmth of the item, the comfort of the item, the formalness and the festiveness, okay? It turns out that the most discriminative features are formalness and warmth. And what we're doing here is we're then looking at trying to predict people's value from a linear combination of these features, okay? Just like we did before. So we fit a model that is basically integrating over fitting weights to each feature that then allows us to predict a value. And we can do that separately for each context, okay? And what you're sort of seeing here is that there's a strong positive feature assigned to formalness, no surprise, in the job context, right? You should look more formal when you're going for a job. And less weights assigned to in the ski or island, okay? In fact, negative weights. Versus in the ski trip, you are assigning, most people are assigning a positive weight on warmth, right? And it's kind of the opposite when they're going to the, sorry, that's what whited out, apologies, that should be down here, that should be for the island, okay? So there's a negative weight on island, and so on. You're kind of not that moving, it's kind of pretty much neutral for the job, okay? And these other features don't discriminate well between contexts on average, but there's a lot of individual preferences as well that vary, okay? Now, what's also interesting about this is I can take these features and I can train a model that has just the high-level features, okay? Or I can train a model that has both high-level and low-level features. So in kind of dark here is all features, high and low level, and here is just high level. And what you're seeing is that the high-level features are based, this is performance of the model, correlation with value. The high-level features are basically doing as well as you can possibly do. And if you just have low-level features by themselves, they do all right, still better than chance, but they're not doing as well as the high-level features. So again, it's suggesting that just these high-level features alone is enough to capture the variance in liking ratio. Another really interesting thing is that what I can do is I can also fit a model. Actually, that's shown, yes, it's shown you that. So I can fit a model to the ratings for the female gender. And then I can apply that same model, like the weights from that model, to the male gender and vice versa. Okay, that makes sense. So, and what I want to do is to see if I train it on the female gender, does it generalize to the male gender on average, okay? And if you have a model that has high-level features in it, it generalizes very well across genders. But a model that only has low-level features basically is no better than chance. So that's also telling you why it's useful to have these high-level features when you're kind of making judgments about things, that it maybe generalizes well across different kind of contexts. We can then do the same thing in the brain to look for representations of high and low level features. And this is through the ventral visual system, again, the same regions of interest. And once again, we see high level attributes are more represented as you move through the visual hierarchy, and low level features are less represented, okay? So you again, this kind of hierarchical organization, again, no surprise. Now, the big question that we're interested in when it comes to the brain for this study is, are these feature representations, say of comfort, formalness, warmth, are they represented in value space or are they represented in sensory space? Okay. So if they're represented in sensory space, they shouldn't change as a function of context. They should just be the same. Warmth is warmth. A warm jacket is a warm jacket, whether you're in the ski context or whether you're in the desert island context. Okay. But if those features are being represented in value space, then those representations should change depending on the context that you're in. And the question you're interested in is, well, in the, say, prefrontal areas, are those feature representations already in a value space? They're kind of like a marginal value for that feature, which will then change depending on context. Or are they just objectively representing how warm the thing is? Okay. So what we did was we did a thing called a generalization analysis. So we basically trained a classifier to distinguish, say, warmth, high versus low warmth, or high versus low formalness, and so on, in one context, like a job, say, or island, or so on. And then we tested it in a different context. Okay. Now, if the classifier still performs well and generalizes well, it means that the representation is the same. The code being used to represent it is the same in these different contexts. But if the performance drops down markedly, that means it's a very different type of representation. That makes sense in these different contexts. And that doesn't mean it's value, but it means that there's a modulation going on between the feature representations as a function of context. So here in this example, we're training in a job context to distinguish formalness, high versus low formalness, okay? And this is our performance, okay? And it's better than chance, okay? And this is in the visual cortex. And what you can see is that it's generalizing well, actually, across different contexts, okay? So if you train it on job and you test it on ski or island, it's doing okay. Same thing for warmth. You train it on warmth in a ski context, it generalizes well to the job context or the island context, okay? Oops. Now, here we're training on formalness, and this is this is not in visual cortex now, we're looking in lateral prefrontal cortex, okay? And what we're seeing here now is we train it on formalness in the job context, and then we tested these other two contexts. There's actually a big decrease in the accuracy. So it suggests that it's representing things very differently depending on context. So the lateral PFC is incorporating contextual information. And we also see the same thing in medial prefrontal cortex. And analogously for warmth, we train it in warmth in a ski context, and it doesn't generalize well to the island or job context. So there's a difference in performance as you go across these different contexts. So what that means is that these feature representations in prefrontal cortex are being modulated based on the context that you're in. And we can also basically look at the value signals now, turn into the value signals. And we're saying, well, now we know that the integrated value is an integrated computation weighted over these different features, okay, that have been trained on each person separately. Or we can have kind of marginal value, which is basically the marginal weight assigned to, say, a feature such as warmth, which will then give you the value of warmth, okay? And what we're looking at here is the average representation of the feature values in red versus the integrated value in green. And what you can sort of see here is that there seems to be this topography in VMPFC and orbital frontal cortex, whereby you go from these more feature value signals, maybe more ventrally, and as you move more dorsally, you get this integrated subjective value. So it's a kind of suggesting that there's kind of feature representations are then integrated into into value even within the ventral prefrontal cortex. So to conclude, what I've shown you is that value signals are maybe actively constructed in the brain for flexible behavior. A value of a stimulus can be in part predicted from underlying attributes in a lawful way. And food as well as art can be predicted from both visual features as well as more abstract features such as taste and nutrition. And the value of stimuli depends strongly on our current goals. It's modulated by context, and that modulation appears to happen not in visual cortex, which suggests that the visual cortex is representing the objective feature, okay? But in the prefrontal cortex, it's modulated as a function of what context you're in, what goal you're in, which suggests that it has a kind of it's a value-like computation, which then we provide evidence that that's happening in the prefrontal cortex. And I will conclude. I would thank all of the people in my lab who have contributed to all of this work, and I get to come and talk about it while they get to slave away in the lab, and I appreciate them. And thank you for your time.
Thanks for the great talk. We've got some time for questions, please.
Thank you so much for the great presentation. My name is Dongwon Lee. My question is, I think you provided like 3 examples: the food example, the clove example, and the art example. But in my opinion, I think that food and clothes example can be kind of like goal directed, because if you don't eat food, you're going to be hungry. And if you like pick the wrong clothes, you're going to be cold or warm. So like I understand that how this depends on context. But in the art example, like artistic stimulus, I wonder how they like differ by context, or if they have like an effect on how you value those, like aesthetic stimulus.
Yeah, it's a good question. I mean, I would say, so imagine I were to say this work of art is worth $5 million, or this work of art was painted by a child in kindergarten, okay? So that will probably modulate your value. And in fact, there's evidence to show that it does. So you kind of, so there are ways in which I think context will also modulate, you know, your opinion of art. So obviously, if you go and see, there are examples. There was a TV show that was in the UK many, many years ago, where they had somebody who was like a painter and decorator who was cosplaying as a modern artist, and he would like flick paint onto kind of a canvas. And if it was presented to people who are supposed art experts, and they were obviously fooled if they thought this guy is an artist, they thought, you know, it's great art, right? So I think that there are definitely ways in which context will also influence your value for art. I mean, we'd like to think it's more objective than that, but I think it's probably not. Yeah, yeah.
Thank you so much.
Sorry, are you taking the questions, or should I just point to people as I see them come up? You want to handle that, or no?
Either way, yeah.
You will. Okay, I'll let you do it. Sorry.
Okay. Question.
Thanks for the insightful talk first. And I wonder that could there also be the neural representation of interactions between the features like high features and low features? Because as I understood, I suppose that the high level features can be the combinations of the lower level already, but I just wonder that how combinations of high and low level features might jointly modulate or subjective value directly.
Yes.
Thanks.
It's a great question. I think, you know, if I can reinterpret your question, I think what you're asking is kind of nonlinearities, right? So it's combinations of things that are more than the sum of their parts. And we did look for that, actually. So there's a whole bunch of different ways we did that. One was to use a convolutional neural network to basically input each image, and then look at to what extent it's integrating over features to compute liking ratings. And it gave us a little bit of an extra predictive accuracy, maybe by a small percentage, but it wasn't huge. So the answer to your question is almost certainly that stuff is happening for sure, those kinds of nonlinearities. And you saw that with the model that I showed you last for predicting food. that this model that had these nonlinearities actually did a little bit better in predicting value. So it suggests that there is these nonlinearities, but we haven't really understood them yet. But I think it's a very important question to understand that it's clearly not just a linear process, but the linear model does so well that we've kind of just stuck with that for now. But for sure, there's these nonlinearities that will include interactions between low and high level or between two high level features. And that's most likely to do, well if we have the right model. The challenge is that we can predict, I guess, correlations of like 0.4 to 0.8 in some people between their ratings and the model predictions. But that's still, that's maybe what, 20% to 60% of variance. Mostly it's around the 20%. So there's a huge amount of variance in people's liking ratings we're not predicting. And I think the other aspect of that is going to be more subjective things, there's going to be semantic information as well that's relevant, which we haven't touched on. Like, for example, if an image has a dog or a person in, we did a little bit on a person being in, that does actually predict value to some degree. But there's also things like your own memory, right? Maybe you like a particular food because you had it when your grandmother gave it to you when you were a child. Those are things which we obviously can't predict easily just from liking rate, from ratings like this. But I'm interested in trying to maybe more individualized. If you could take a person and just get huge amount of data about their liking ratings and their history of memories, and and maybe we can do better in predicting their individual preferences. I think that's a really important direction, which gets also towards kind of nonlinearities too. Yeah.
Question over there.
Thank you for a great talk. I was actually wondering about how the actual integration computation between individual feature representations would look like in terms of neural circuits or neural systems and so on.
So, I mean, that's a good question. I think we don't know the answer. I mean, one idea, simple idea, would just be, there's one set of neuronal population representing an individual feature, and there's connectivity, synaptic plasticity between that and the value signal, and it's basically just a change in that coupling is setting weights, different weights to features, which maybe are established, through experience, through as you experience different foods and learn about them or experience different images. So that's the simplest possible model that you could have. That would be, my first place to start. But I think we're going to hopefully have more capacity to look at that at the single unit level, because we can look at those kind of connectivity between neuronal representations of features and see how that's, how that plays out. So that would be one kind of very simple, kind of simplistic model. But I'm sure there's much more complex and richer ways to think about it.
Question.
Thanks for the great talk. I have a question about what would happen after this value construction is completed. For example, if people are habituated to the point where they forget about these low-level, high-level features, they just like it. And when that happens, and then you ask people, why do you like it? Is it because it's tasty, or is it because it's healthy? Or some other cases, you can, they might have to, for some reasons, they might have to update their value. So when that happens, during which time, do you think this causal computation will be reversed, starting from the value representation and then retracing back to this high-level feature representation and low-level feature, just to update your value construction process?
Yeah, I mean, that's a great question. I mean, I think obviously what the machinery that I've not talked about is probably this machinery that's representing context. We actually were kind of trying to do that in the last study, and we couldn't really decode very reliably a kind of context region. So that's still a very open question. So what I would hope to see or expect to see is that, at least for these kind of very high level things like, food, clothing items, for example, that there's maybe some representation of the goal, the overall goal, which is maybe hierarchically represented, which we know from other work is the case. And that representation will then inform the value of the individual, depending on the goal that you're in. Let's say you're in the shopping goal, sorry, the island goal, or you're in the, ski goal. That is then going to modulate probably exactly how it does it. I'm expecting it's going to modulate the features themselves, feature values, which will then feed into value. So I think rather than kind of value going, the causal chain going from value back to individual features, I think it's the goal representation, the overall goal, behavioral goal, which will then modulate the features. That's what I'd expect to happen. You also asked about, like if you have this maybe habitual-like process. And that's a very interesting question, because you could have, rather than actively modulating the value of the features, you could have different feature weights in different contexts. So like, I don't even have to compute or change my value computation. I could have just like, when I know I'm in context A, I know I like coffee. Morning time, I like coffee. Afternoon, I don't like coffee. And I've just learned that so much that it becomes like a habit-like representation. So that's also possible. And that would be very interesting to look at how that changes over time. But also when it comes to things like food, I think there's probably something more fundamental going on, which I expect that those things are very represented in subcortical areas like the hypothalamus, that cares about thirst or hunger. And you're probably going to have this also similar type of modulation of the, these feature weights, like how much am I going to value fat, or if I'm like I have a salt deprivation, I'm going to value salty things more, right? And that's going to be, I think, a bottom-up kind of type of representation from these very, very kind of elementary parts of the brain that are encoding these motivational states, I think. So I think it's going to differ depending on the type of computation. Yeah.
Professor.
Hi, thanks, John, for the great talk. I have a question about individual differences. It might be related to Sangwan's question. Like overall, many people may prefer, may rate the taste or healthiness of the food, as an example, but some people may prefer other features more. Yeah. And like for drawing, some people may like this picture, but some people may not like this picture. And like some people may care more about higher order features, but there might be other people who might care more about lower level features.
Yes.
Have you looked at such individual differences?
Yeah, I mean, it's a great question. It's something I'm very interested in. I mean, I think it's interesting from both from just a general question about are there cultural variations? Like right now, the people that we had in the lab on the food study and from the art study are from LA, okay? But let's take the food example. I think it's more useful. But there could well be big individual differences, like if we test people from different cultures, in terms of, and also from, for example, how much, as you said already, how much they value healthiness. I think that there's almost certainly going to be a lot of variation. We haven't looked at that yet, but it is on our list of things to do, to maybe look. Fortunately, with online studies now, we can test people across different cultures. So I think it'll be interesting to do. And I think the other interesting question is are there changes in how people integrate these features that might relate to different disorders? I think, for example, eating disorders is an obvious one, or obesity. Maybe there's going to be differences there. And yeah, I think it's a very interesting question, and it's something that we want to kind of do. In the art, what we do see is that, you saw, remember, and I showed you that image with that figure with the variation across people and how well the model could do. That's based on an average model. But if you actually look at people's ratings, there are different clusters. Some people like abstract art. Most people like very concrete art, like a very veridical painting, realistic painting. And there's actually about 3 different clusters of people's preferences in art. And if the model that's trained on the average one is not going to capture that variation. So I think you're absolutely right. There's a lot of variation across people. Yeah.
Question over there.
Hello, thank you for your interesting talk. I have a question about your organization of the features. So I could understand that visual stimulus can be a really basic features, but how did you order that? Like, for example, in the food example, like there is a nutrient feature. So like, how did you really order that features from the low to high level?
Yeah. Well, I mean, I think the answer is, well, actually, that's a great question. One way to do it that we started was just based on what we think. So, you know, we just said, we didn't really say there's any hierarchy within nutritive features. We didn't say there's a hierarchy there. So we just said, let's take a bunch of features and see how well they predict behavior. And then you can take the subset of features that are best predicting behavior in a model, you know, select from those, okay? And we then said, well, let's take taste features and let's see how well they predict. And then we said, let's put them together in a model and see how well they do. And we can't say anything about the order there necessarily between, say, taste and nutrients, but that's where we can probably go into the brain. And that's where I want to use techniques with higher spatial and temporal resolution to try and see, well, which ones are represented first. And we obviously expect the visual features first and then these other features, and the order will come from what we see going on in the data. So that's one answer. The other thing is, you know, how do we know whether it's high and low level? I didn't present that, but this was work that actually Sang-Yoon Yi, who was a graduate student in my lab, did, was we trained a convolutional neural network, and we didn't actually train, the network was pre-trained. It was one of the off-the-shelf convolutional networks. And then we trained it a little bit on doing liking ratings. And we could then look within the network at what layers are representing these different features, say for art. And you can sort of see, actually, I didn't present it, but I can go back to it now since I'm here. Yeah, so we can sort of see something that kind of looks like this, where we're trying to decode from the network these different features. And so hue is decoded best from early layers. These are different layers of the network. Concreteness is decoded best in the higher layers, and so on. So we can actually use that to characterize the features. And we had one feature, which was the presence of a person, which we initially labeled as low level, but actually it was high level because it was actually more represented in the higher order layers. So that's another way you can actually do it. Yeah.
Do we have any questions online? Okay. All right. Thank you so much again.
Thank you.
Okay. Our next speaker is Professor Woo Young-an from SNU. He's going to give a presentation titled Naturalistic Paradigms and Real Words for Unveiling Neurocognitive Mechanisms in Decision-Making and Addiction. Take it away, please.
Today, I'm going to talk about and try to convince you why using naturalistic paradigms would be useful to translate research into clinical practice. This is kind of a mission statement of my lab, which actually you can find from my website. I haven't changed this figure for many, many years since I set up my lab. And as a clinical psychologist, I'm a clinical psychologist by my PhD with some background in engineering and applied physics. What I want to do is I study transdiagnostic phenotypes like impulsivity, given the limitations of the current DSM and ICD diagnostic systems, and use computational neuroscience or computational psychiatric approaches and try to come up with some essays or paradigms or tasks or essentially decision-making processes that can be useful to study the phenotype that I study, and use the best machine learning or artificial intelligence algorithms to develop some biomarkers or surrogate markers that may lead to some innovative treatments or assessments. So this is a statement, mission statement, not just for me, but for many clinical researchers. However, it has been really challenging to translate research into practice. And I'm trying to convince, and also I'm going to show you some series of studies that are being conducted in my lab that try to better understand these decision-making processes using the state-of-the-art paradigms and approaches. So we often use the brain, the black box as an analogy for brain, and we use computational psychiatry approaches. And there has been many workshops, meetings, conferences, and like a lot of funding has been put together, and efforts have been put together to make it come true. But there have been many, many challenges. These are some of the challenges that we need to overcome to use for using computational psychiatry approaches and to translate research into practice. And I'm going to focus on this last challenge in this today's talk. And I'm going to show you some. I'm going to share some studies that use some naturalistic paradigms. And I'm going to give you some updates for those who have already seen this paper talk before, with some updated findings with some real rewards in the MRI, which I'm really excited about. And if you actually think about impulsivity or addictive behaviors, what are the things that occur to your mind? These are the images that occur to my mind. So I use these pictures, like jumping off the cliff or using some illicit drugs that may be illegal or that may even kill your career. And how do you actually measure impulsivity as a clinical researcher or any researcher from any researcher's perspective? The easiest way is just ask questions or just use surveys. And these are the actual items that I got from a impulsivity survey. Like we can ask, like, am I self-controlled or I act on impulse? However, there are certain limitations. Like I can even easily lie because I don't want to reveal that. Some people may want to reveal that they might be impulsive. So we also use behavior tasks in the laboratory, like a go-no-go task or stop signal task or delay discounting task. And these tasks often ask participants to press a button or withhold from pressing a button. So we have been talking about the importance of context. And as you actually have already learned, this kind of behavior might be relevant. You are in a context of like in a simple, like a chamber. Like if you're a rodent, where you have nothing to do but pressing a lever or not, this kind of behaviors or tasks might be relevant or might be very appropriate. But decisions we have to make everyday life are a lot more challenging and complex. Like, should I come to this symposium or not? Should I do it via Zoom or should I come here in person? Like for those in grad school, should I go on for a postdoc or should I go to industry or should I like in so many decisions you have to make in terms of relationship too. So people have been thinking like, these traditional paradigms, although they have been extremely useful, it might be better to come up with some more real world like tasks or paradigms. So there has been a lot of interest in using more real-time or naturalistic paradigms to better mimic the real world behaviors. And these are some of the review papers that you may find interesting. So in my lab, I also feel that these tasks might be overly simplified to translate the data from the lab to make predictions in real world. So we have been, my students and my lab mates and I have been, and my colleagues have been trying to come up with some naturalistic paradigms that can actually allow us to make better predictions for real world behaviors. And I'm going to show you some of the studies, and I'm going to show you also some of the puzzles and mysteries and gaps that we want to address with this line of research. So the first study is a driving task. So the gap or mystery that we want to address with this study is that when we assess a construct like impulsivity, let's say, as I said before, we can measure impulsivity with questionnaires or behavior surveys or even with neuroimaging. But impulsivity when assessed with surveys or behavior tasks, they don't correlate. When we actually check their statistical relationship, their correlation is not often significant at all. And people have been speculating why. Some people think it's because they measure different things. Questionnaires measure more stable trait measures of impulsivity, but behavior tasks might measure more like a state features of impulsivity. However, many researchers in this room may want to claim or may want to assume that those tasks or paradigms, they should also measure stable characteristics of individuals. And that's what we often assume. Because if we think those tasks are only like a state-like features of an individual, then why do we have to care? We often think those behavior measures and computational markers that we can derive from those behavior data are presumably stable over time. So what we speculated or what we hypothesized to address this gap was probably this kind of a low correlation between surveys and behavior tasks might be due to the too simple nature of a behavior task, because those tasks, whether press a button or not, might tap into the constructs that are assessed by the surveys. So the first task we came up with was a driving task. So this study was led by Sang-Ho Lee, who was a postdoc in my lab, now at KAIST. And the reason, as you may heard before, we chose this driving task is because this is a perfect manifestation of our true personality often manifested on the road. So, however, rather than just making it too realistic, we just made it a little bit easier and simplify to make it easier to measure their impulsivity on the road. So what participants have to do is drive this green car as fast as possible to get more bonus money without crashing into other cars. So this car just crashed into other yellow car and lost quite a lot of money. So we thought, okay, this is a really cute task, and this may reveal some characteristics of an individual. And what I like about this task is participants don't get bored. Like if we ask them to complete some traditional laboratory decision-making tasks, they often get very easily bored, or they even fall asleep. But with this kind of task, they sometimes even want to play more. But the challenge is, we're going to get a lot of data. We're going to get a lot of real-time data, a lot of features. So we can't use a traditional computational modeling approach. So what we did was we collaborated with an expert in AI and reinforcement learning, Min-Hwan Oh, at SNU, and we applied a deep neural network-based inverse reinforcement learning that can allow us to generate the real-time reward functions at each state and action during the highway task task. And this is an illustration why we shouldn't just use the optimal agent like the AlphaGo or deep-Q learning. So So if we actually use a deep-Q learning, which is an optimal agent, this is what deep-Q learning performs on this highway task. So the performance of this deep-Q learning agent is so much better than human agent, what humans can do. So the point is, we can't use this kind of optimal agent for characterizing the individual differences on this driving task. And I think our study is one of the first ones that actually use the deep neural network approach to characterize the individual differences and measure the impulsivity in this way. And the finding is here the y-axis is the survey score of impulsivity with the BIS, something called BIS, and x-axis is the impulsivity score from a traditional laboratory task like delayed discounting rate or go/no-go error. And as you can see, there's no significant relationship between surveys and laboratory tasks. However, with the highway task, there's a significant relationship between survey and the highway task performance. And this figure shows that this inverse reinforcement learning approach that we use is doing a decent job in making the actual behavior. Although it's not perfect, it's actually tracking the patterns of the human behaviors. And I think this is the most interesting finding here. The passive overtaking is, the green car is passing the yellow car just without changing the lane. But here, in the case of active overtaking, the green car is just behind this yellow car and maybe annoyed by this yellow car ahead of the participant. So change the lane and overtake other cars, which is often really kind of a fun moment when driving a car. And here you can see that people with high impulsivity, which is the orange line, shows greater subjective reward compared to people with low impulsivity, which is indicated by this the orange and blue lines. And here's the onset of overtaking. So I think this is great, and it was published in Psych Science last year. And what we wanted to prove further is, okay, these are the subjective reward functions from the IRL algorithms, but is it really represented in the human brain? Is the reward functions really represented in the human brain when driving a car with this task? So we ran a fMRI study using the same task, and we are also currently trying to use a modified version of this highway task where other cars can actually can indicate whether they're going to change the lane or not first, and then they can also cutting in my own car, which is often very annoying when you are driving a car. And here's the findings from the MRI data. First, we, as a sanity check, we first ran a GLM analysis, general linear modeling. And here's the findings at the onset of overtaking and at the onset of crashing into other cars. And as you can see, at the onset of overtaking other cars, there's a bilateral striatum activation, and which was the opposite at the onset of crashing into other cars. So, so this kind of sanity check that, okay, probably overtaking and crashing into other cars are actually represented in the reward center, like or striatum. And this is kind of we kind of did a sanity check. And the another analysis that we did was we divide the brain into 120 regions of interest and extracted the time series of all the ROIs from 120 ROIs. And then we try to predict the subjective reward functions or in data changing dynamically during the whole driving task. And we try to identify what are the regions that actually contribute to the predictions of this subjective reward during the driving behavior. And we found many different regions, including striatum, dorsolateral prefrontal cortex, and parietal cortex. And we are currently writing up the paper. So we are pretty excited about this highway task. So we are currently coming up with some other more naturalistic paradigms that can also really good mimicking the real world behaviors. And one of the paradigms that I'm also really excited about is this crosswalk or pedestrian task. So because not everyone drives here, but everybody actually is a pedestrian. And I want to highlight that this kind of behavior of a pedestrian or an individual is not the real behavior. It's actually a simulated data from the agent. And the findings are pretty exciting and promising. So I hope I can present the findings maybe at another occasion later, which is led by Jin Woo-jung in my lab. And the second study that I wanted to share is a study using a video game called Minecraft. And why this is an important approach I'm going to explain a little more. And we try to address the issue of exploration versus exploitation bias in addiction. So, exploration versus exploration is an important decision-making strategy. You shouldn't exploit too much or you shouldn't explore too much. You have to have a good balance between the two strategies. And there has been some studies that examine this exploration versus exploitation bias in addiction, but the findings has been kind of a mixed. Typically, what researchers use to study this bias is something called multi-unbanded task. I'm going to call it MAB task. So here, there's like 4 different options or bandits, and the rewards for each bandit is fluctuating over time. And so you have to learn which option is the best option on each trial and try to find the best option and try to maximize the cumulative reward as much as possible by adequately balancing the exploration and exploitation. However, even though this task is also really, really useful, there has been some limitations. So this is a map of like restaurants here at SNU, and the limitation of using this kind of simple task is that, let's say you want to do some exploration or exploitation for your lunch or dinner, but then there's a lot of factors you have to consider, like a distance or how much time it's going to take from the place where I am, and what's the price, etc. And there's a lot of uncertainty for doing some exploration. However, using this kind of simple 4-unbanded task may not have enough ecological validity that we may as wish. So, and as I said before, the cost, like time and effort for navigation, can't be really considered for making choices on this simple task. And as you may have done for your trip, for a trip for lunch, for many of you, you actually have made a lot of locomotion and visual exploration. If it's your first time visiting SNU, then you might have been drawn to a new features or kind of novel scenes on campus. And some of the locomotion activities and visual exploration might be irrelevant or might be actually interfering with your goal-directed behaviors or the goals that you actually have to achieve. So, however, with this kind of simple button pressing task, it's not easy to examine all those features. So what we did was, in collaboration with Wonmok Sim at Sungkyunkwan University, based on the 2D or traditional multi-unbanded task, we used a Minecraft game where where participants can actually explore or exploit among 4 different colors of flowers within the Minecraft game. So I'm going to show you a kind of screenshot, like a small video clip, a short video clip, how it's going to be done from a participant's perspective. So as you can see, a participant can actually pick flowers and go around and decide, okay, what's the best flowers, what's the best color of flowers that I have to choose on each trial. So, by the way, this work is led by Hyunmin Lee in my lab. And she recruited not just healthy controls, but 54 smokers. But importantly, the smokers had two different groups. One smokers came to the lab after they had been smoking as usual. And the other half of the smokers, they came to the lab after 12 hours of abstinence. And the reason is smokers often behave or make decisions differently based on their abstinence status. And why do we study smoking? Because smoking is one of the most prevalent disorders, most prevalent substance use disorders in the whole world. And not sure if you know, but this is one of the most difficult substance for quitting. Probably other than heroin, because there's so many components and elements in nicotine, and the companies that don't want us to quit. So, and they have been able to, make it possible to add so many components and elements into the cigarettes. So the procedure is every participant perform both the traditional 2D multi-and bandit task and the 3D multi-and bandit task. And the order was counterbalanced, and they also complete some surveys. And we model their behavior with some computational models. And importantly, there's two important parameters that we want to assess across the groups. The first one is the exploitation versus exploration parameter, how much you explore or exploit. And the other parameter is the cost sensitivity, how much you care about the cost for navigation in the Minecraft environment or context. And as I said before, we are able to also collect their locomotion activities and visual exploration behaviors during Minecraft game. And the modeling findings indicate that with the 2D multi-and bandit task, we found no group differences either behaviorally or model parameter-wise. However, with the 3D, the Minecraft-based multi-and bandit task, we found that abstinent smokers stand out compared to the other two groups. As you can see here, the abstinent smokers showed greater exploitation. In other words, they exploited what they think is the best a lot more than healthy controls or steady A smokers. And they also showed reduced cost sensitivity compared to healthy controls and steady A smokers. We also found that the visual exploration features also contributed to for discriminating, classifying the abstinent smokers compared to other two groups. And in other words, abstinent smokers made more distracted or disorganized visual exploration. Like they move around their heads a lot more compared to other two groups. So what are the findings? So with the approach using a naturalistic paradigms, we found that the Minecraft-based multi-and bandit task may have greater construct validity, and it may better mimic real-world behaviors for exploration or exploitation. And we found, again, the abstinent smokers show greater exploitation, and reduced cost consideration. And we found that, we found some evidence that the motions and head movements that we can actually collect from this kind of navigation task may contribute as a marker, or may be able to, might become a potential marker for a psychiatric condition. And here's some new findings with the real rewards in the magnet. Here, this study was led by Jang-hyun Lee and Eun-hui Lee, and this was work done in collaboration with Josh Brown at Indiana University. And the gap that we want to address with this task is there has been some debate about reward processing in addiction. Like has been some theory like incentive sensitization theory that drugs are processed differently compared to monetary, even other primary rewards like food and sex. Or other camps or other findings suggest that that's actually not true. There's a more like a general reward deficiency or anhedonia among substance users. So it means like among substance users, they show down-regulated responses to all the rewards, not just the substance they use, but all the rewards, including money and other primary rewards. So this kind of a model has been pretty hard to be tested in humans, because in humans, it's really hard to provide primary rewards or substance they use, especially inside the MRI, because it's not for some ethical reasons and some technical reasons. You know, what are the way? It has been very difficult. But with Josh Brown, who actually made the MRI compatible vaping device that can allow us to provide e-cigarette or e-cigarette vapes inside the MRI in real time based on their decisions, we were able to actually provide nicotine rewards, not just the monetary rewards, while they make decisions. So we ran some pilot studies, and it is my hope that with the new MRI and with the new MRI and with the facilities, we can have a lot more enhanced signals and a lot of other innovative studies with this kind of vaping devices. But this is the findings of the first study of the vaping study. So the first thing that we wanted to do was we first wanted to identify or validate the reward and risk signals with healthy controls and smokers. So for that, what the task that we use is something called, like, I just called it a Prusov task, because the Prusov is the first author. Not sure what's the name of the real task. Maybe John may know, because this was published in CATech back in 2006 and 8. Well, what's nice about this task is with the single task, we can actually examine not just the reward and reward prediction error, but also reward risk and risk prediction errors. Here, we use a model called mean variance model, and the risk is defined as the variance of the reward outcomes. So we recruited around 44 healthy controls and 22 smokers. And what we did was, with health controls, we only gave monetary rewards. But to smokers, we gave both monetary rewards and also nicotine rewards. And we first wanted to check their craving level. So this is before experiment, and this is after experiment. And this yellow line is when we gave monetary rewards, and the red line is when we gave vaping rewards. So as you can see, when we gave monetary rewards after the experiment, about an hour after the experiment, their craving level went up, but it was not the case for the vaping reward participants. And this is the fMRI signals related to reward prediction errors. With healthy controls, as has been well known, we found bilateral activation in the ventral striatum with reward prediction errors, which was smaller, but still we were able to see some small voxels or blobs in smokers with monetary rewards. But we didn't find any evidence of intact representation of the reward prediction errors when we actually gave vaping rewards to smokers. And what about risk? It has been well known that anterior insula is encoding risk prediction errors, and we were able to replicate that with healthy controls. Oh, sorry, the group name is missing here. But these healthy controls, showed intact anterior insular responses to reward prediction error, risk prediction errors, but it was not the case for the other smoking groups. And we also draw some time series of the risk prediction errors, which is, this figure is from the Prushoff et al. 2008. And these are the figures from our data. So with healthy controls, the red, orange, and green is high and medium and low risk prediction errors, and which was pretty evident in healthy controls. But in smokers with monetary rewards, the high risk was only separated from the other two conditions, but it was more smooshed, and it was not really evident, clearly distinguished between the three conditions with the smoking and vaping condition. Of course, the sample size is still small, and we need to recruit more, and we need to replicate it with another study. But these studies highlight that there's something special about the real rewards. The second study is led by Jung-hyun Lee, and this experiment is to assess the context-dependent subjective values in a two-arm-based decision-making task. So here, participants made decisions between two different options, and the difference of the expected value between the two options varied across the pairs. And we also recruited healthy controls and smokers with monetary rewards and nicotine rewards. And again, when we measure their craving level before and after the experiment, with the money condition, their craving level went up, but with a vaping condition or vaping rewards, their craving level actually went down. So there was a significant interaction effect of the timing and the type of rewards. And behaviorally, we found that smokers showed worse performance compared to healthy controls when the difference of the expected value is the biggest compared to between the two conditions. And when we fit the behavior data with many different models, we found that a model that actually weighs expected value and expected frequency of rewards is the best fitting model. And here's the equation for the best fitting model. So the Q value for the action of each choice is the weighted sum of expected value, or EV, and expected frequency of reward, which is EF here. So if the omega is close to 1, that means the person is heavily weighting on the expected value. But if the omega is close to 0, that means the person is only caring about the frequency of the rewards. And there's a simple reinforced learning rule and softmax rule for making actions. And we compare the model parameters of the healthy controls, smokers with monetary rewards, and smokers with vaping rewards. As you can see from these figures, smokers with the vaping rewards are standing out, are separated from the other two groups. So only the smokers with vaping rewards showed reduced learning rate compared to the other two groups. And the omega, which is the weight for expected value, was much smaller compared to smokers, compared to healthy controls, and even compared to smokers with monetary rewards. And we are still analyzing the fMRI data, but this is the fMRI results when we compare the vaping rewards minus monetary rewards. And we found significant activation, greater activation in bilateral striatum or putamen and SMA compared to, which is greater for vaping rewards compared to monetary rewards. This is, of course, this is only among within the smokers. So what do these findings indicate? I showed there's two different hypotheses or views of the addiction and reward processing. And I think our findings rather support the first one, that drug is treated differently compared to other monetary or other primary rewards. Of course, these are preliminary findings, and we are currently doing more analysis and plan to collect more data with the new MRI facilities. We are also using some movie watching paradigms, which is in collaboration with Monica Rosenberg at the University of Chicago. So we show movies instead of some pictures of drug cues, because cue-reactive paradigms is one of the most widely used paradigm in substance use disorders, and people typically have just presented pictures and images, visual images, to participants to evoke their cue reactivity or cue-based craving. However, there's a more and more interest in using more naturalistic stimuli. And in this study, we presented movies, video clips of alcohol drinking that we actually got from a lot of Korean like yenim or Korean dramas and so K-pop dramas. And we find that the craving is actually modulated by how self-relevant do you think the stimuli are, and we also found that the videos evoke greater craving compared to just the pictures. So I'm not going to show you a lot of details about this study, but we are also using expanding this line of research to nicotine smoking, and we are also preparing for a study using video clips of nicotine smoking in a new follow-up study. So I think this kind of highlights for the utility of using naturalistic paradigms. And this is the picture, the mission statement that I showed you earlier. And you may notice what's the difference between the picture or the mission statement that I showed you earlier and this one. It's this one. So instead of just the decision-making processes and what's the balance between decision-making, some of the decision-making subsystems, we think using this kind of naturalistic assays and paradigms may allow us to come up with better biomarkers and better surrogate markers, and which may, in the end, lead to make us better treatments and assessment, and make it a little more feasible to translate research into practice. And lastly, and most importantly, we need good mathematical models for the mind. I think some of the other presenters, including Jiuk Cha, presented some novel approaches using AI, and not just the LLM type of model, but there's a new paper that uses actually the recurrent neural networks, and there are some other new approaches that also combine like traditional computational models with some machine learning models. So there has been a lot of interest in building a model that can actually translate across multiple contexts and tasks and make predictions in a more generalizable way. So essentially, we need to build a model that can predict and simulate human behavior in any domain, but this is a kind of a big question and big mission for many of us, and hope we can actually make it work together. So I think that's it for my talk, and thanks so much. And thanks for all the support from NRF and NIH and Ministry of Health and Welfare in Korea. So thanks so much.
Thanks for the wonderful talk. We have some time for questions. Question over there.
Thank you very much for the very interesting talk. I have a question about impulsiveness project. So the way I see the impulsivity is that you make a choice regardless of your value. Like if there's no reason, but you just want to do it. So in that regard, I wonder if you have a look at the time point where they change the lane, even though inverse RL model doesn't make any predictions about neither positive or negative reward.
Mhm.
So like it's basically you have a zero value, but you just take an action.
Right. So thanks for asking the question. One thing that I want to mention is that impulsivity, people think impulsivity has multiple definitions or dimensions or domains. So I think you are correct that some people may make some impulsive action without thinking or without thinking about action, the value of the option. And I think that's kind of a response inhibition kind of a dimension. But some people, there's another dimension like, immediate versus delayed rewards. And this is actually comparing the different values. Like I care more about immediate reward compared to the delayed reward. So in that case, people may also care about values. So it really depends on how we actually define the impulsivity too. But I see your point, and in this highway task, we only provided some evidence of different manifestation of the impulsivity in this context. But I agree with you that there's so many other different analysis we can do. So like, changing into other lane without really thinking about any without considering the value at all, it can be it can be kind of a exploration versus exploitation too. So I wish we can actually differentiate that exploration, exploitation from other strategies or domains, and that's one thing that model can incorporate in the future too. But there's so many things we need to do too. Yeah.
Hi, great to see that new data on the nicotine stuff. Very interesting. So I was just wondering on the risk reward task, are they more risk seeking in behavior for the nicotine, the rewards?
The first one or the second one?
I think the first one.
The
yeah.
push-up one.
Yeah, push-up one. Yeah. Or did I miss that? I'm just behaviorally, are they more
Behavior.
Yeah.
So this is a task where you are supposed to see no behavior differences among individuals. I mean, this is so in
Oh, that's right. Yeah.
in
'cause
this
there's no-
task,
yeah.
it's like there's a one car and the second car, and then
Right. Yeah. There's no behavioral.
yeah, there's no
index. Yeah,
behavior.
sure.
The only behavior we actually collect here is that you actually have to indicate whether you actually won or lost. And it is presumably like, it has to be like close to 100%. But actually, I recently checked the data with Eunhui, and to our surprise, the accuracy was not that high, but at least there is no group differences among the three groups.
Right. Sure.
Yeah.
Yeah.
That's one of the things I like about this task too. There's no differences in their behavior performance, so it's not tied to their behavior performance.
Question?
Thank you for the fascinating talk. My question might be very detailed. It's about the craving levels between the monetary reward and the nicotine reward. I wonder, like for money, there is no limit to having feeling satiety, like you can get more money, but for nicotine, there's a level of being satiated. So I was wondering, like, why did you compare the craving levels between these two rewards? And maybe if you find a different reward that has like a limit to it.
Right. Thanks for clarifying question. So the craving level is how much they have craving on for smoking. And as you may recall, they came to the lab after at least 12 hours of abstinence. So they are dying to smoke a cigarette. And we thought, after one more experiment, which is presumably very tiring, their craving level should go up because they have not been able to smoke for additional 1 more hour. So that's what we observed with the money condition. And with the vaping condition, they smoked a little bit, although we didn't allow them to smoke too much on each vape for the reasons that you can imagine. So, but they were able to smoke more, quite many times. So their craving level decreased. And this is kind of a sanity check that they must have smoked, during the experiment. Yeah.
Thank you for the clarification.
Question over here. Professor Lee.
Thank you. I think this is related to a couple of the questions that were already asked. And I think about the definition of impulsivity and what that means in this case. I know you're trying to come up with like a task that could correlate with the surveys, but at the same time, we've been discussing also with John's talk, the context specificity of how our brain encodes value, and therefore shouldn't our definition of impulsivity also depend on context, and perhaps the behaviors or questionnaires or whatever it is should also be context specific? How should we navigate this kind of thing in clinical psychiatry?
First, verbally define any construct is wrong. Many people think. Like, if you can try to define what a chair is verbally, can you come up with a perfect definition? Try it. I can challenge your definition. It's probably not possible. Like, try to define what a pen is. It's probably not possible to define what a pen is. Sorry, I'm not trying to go too philosophical at all, but verbally, once you verbally define a construct, there's so many holes and things we actually miss. So like impulsivity, is it really impulsivity? Like driving behavior here, is it really impulsivity, or is it related to maybe anxiety, or is it risk taking? These are all related constructs. But I just try to use the most closely related construct, if I can define what my approach was, and try to measure the construct as precisely as possible, and try to make a more unified set of battery for impulsivity or whatever construct.
Yeah, I think that makes sense, but I guess I'm wondering whether this measures a particular kind of impulsivity that is kind of contextual.
Yes, absolutely. So any decision they make here is depend on the context or environment they are at. I completely agree. And that's why we ask smokers to remain absent for at least 12 hours to put them in the context of abstinence. So if they came to the lab, as smoking as usual, as you saw, even though they are the same, presumably the same smokers, even smokers show very different behaviors when they came to the lab with abstinence versus when they came to the lab after they have been smoking as usual. So context matters, absolutely. And yeah, how we can actually make a quantifiable definition of impulsivity based on the context, that's the thing we need to do.
Do you think this task and the driving task would have the exact same results, or do you think they would have different kind of results? I think that's my question, like task specific access of a certain type of its impulsivity.
I think they are tapping into different things. That's why we use different tasks, I want to say. But yeah, they measure different things. With highway task, I wanted to see how they actually make decisions on while they drive cars. And when they're about to crash and when they're about to, when other cars cutting into my car, they have to make decisions accordingly. And here they have to make decisions, what kind of flowers they're gonna chase, they're gonna go around, and whether they're gonna look around or they're gonna, make some irrelevant motions toward when they move toward a like red flowers. These are all the features that we want to collect. And for now, we only use very kind of a simple type of analysis, but hopefully, even with the same data, I hope we can use some data-driven approaches like a machine learning or some like artificial intelligence or some, so hopefully we can combine theory-based and data-driven, theory-driven and data-driven approaches all together and use kind of a hybrid, kind of integrated approaches.
Thank
John.
Just another question, kind of actually following up a bit on that is, you think if you were to run this task many times, across weeks or months, an average people, would that correlate, or one of these tasks or some combination of them correlate better with the impulsivity questionnaire scores?
You mean repeated measures may help?
Yeah, kind of trait-related average of their score, would that give you better leverage? I mean, maybe it's some combination of these tasks or one of them in particular would
I think
correlate better
so.
with impulsivity.
I think so. I mean, that's one of the things I want to do. I want to make people play these games repeatedly over time because our mind is fluctuating a lot. Like our tiredness, our stress level, our positive mood. Your positive mood may be a lot higher in the morning when you are less tired than 3:30 p.m. By the time you must be very tired and sleepy. So I think repeated measures, which was actually, there's a lot of evidence that repeated measures can actually improve the measurement and reduce the measurement error. And it can also capture the dynamic trajectories of the human mind. So one thing I like about this naturalistic paradigms again is that people don't get bored as much compared to more traditional tasks. So hopefully, we can kind of make people play these games, and they have fun so that we can motivate them play those games more frequently and then use them to use the data from the games to get to know more about those players. So mutually beneficial. Yeah. They have fun playing games, and for us, we get to know about them. Gary.
Really, sorry, to follow, I think, earlier point about the definition of impulsivity. I think that's not quite the same thing as defining a pen. I think action, we can define an operational definition. I think that's what we're thinking about. I think when I think about a game, when I play it, so if someone has zero impulsivity, what should the behavior they should exhibit? I'm not actually quite sure what that is. If you use that driving example.
Right. That's a good question. How can we define impulsivity in the context of highway task? So one thing that I actually thought about in this context where highway task was that if you actually see how the green car is driving, being driven right now. So, this car just changed the lane far much before the yellow car is, the distance between my car and the car ahead of me is quite large in the beginning. But then to maximize your rewards, you sometimes have to make risky movements. You have to rather change the lane just before get crashed into other car, and rather than just change the lane and wait for a while. And so that, you know what I'm saying, right?
Absolutely. So I think actually I can, then the issue is, for example, a person with a faster reaction time, for them, what is taking a risk is
Right.
And when you repeatedly play the game, also I think there's some issues there.
Right. And I think, and I think there might be a data-driven way of measuring or getting a impulsivity score from the real-time data. We actually use more like a handcrafted measures or features. Like we define certain features, like distance from my car and other car, and we use that feature as like a input for the IRL. However, in the future, I hope maybe AI algorithm can actually detect what are the important features in real time on the fly, and they can maybe based on like 30 minutes of driving data from the video games, they can calculate, okay, your impulsive score is 95 out of 100. And maybe that they may need some repeated measurements from many subjects so they can get a distribution of the impulsivity or something like that. But yeah, that's a good point. Yeah.
Thank you.
Yeah.
Any other questions, guys? Okay, thank you very much.
Thanks so much.
It's too bad that one of our speakers, Dr. Eli Merriam, couldn't make it due to the government shutdown in the United States. So unfortunately, he's not here today. So we're gonna skip his session, and our last speaker is Professor Sang-Hoon Lee, who is the director of our imaging center. He's going to give a talk titled The Nature and Dynamics of Working Memory Representations in the Human Visual Cortex.
Thanks for the introduction, and also I also feel sorry for missing Eli. And initially, I thought he's just trying to make an excuse not to come. But I confirmed from our colleague John, to confirm that it's illegal for a government employee to give any talk, and even online is impossible. So I begged him just to put his name and title still. Is that okay? And then he says it's okay. So it's sad, but we can take a look at things from different perspective. We're all tired. Now I kind of thankful. This is my talk, and I have a little bit burden off my shoulder, and then concentrate on my talk today. But to be honest, just Woo Young-an gave a talk, and he stressed that the importance of the making experiments as enjoyable as possible, so they can concentrate. But my experiments are totally opposite. It's so boring. Just showing a little stick and remembered orientation. So sometimes I'm kind of feel bad that my parents are asking, "What are you doing?" And then I describe them. I show students to a little stick and then try to remember for 30 seconds and estimate. Oh, so kind of Jung-Ho Lee mentioned that his son is like, "Oh, you're not studying wire and just covers." Yeah. So what I'm trying to do now is try to make my talk is lower expectation, and it's not gonna entertaining, but I'll do my best. So I sometimes walk around, and so just I'll do my best, but lower your expectation. But again, why I don't moving to which button I should I press? Okay, thanks. So believe or not, we are, according to Harvard's Business Review article, they did a recent survey. Believe or not, we make like 35,000 decision makings a day. That's unconsciously. But more conscious, aware, the number of decisions is about just a statistics of 70. So this number is carries not the meaning a lot, but I want to stress that those daily decision making heavily involve retrieving past experiences. and in response to ongoing demands in current situation. For example, as Korean hiker, because I never been to Canada before, so across Pacific Ocean and arrive in Vancouver, and then I like trail hiking, so I decided to visit Stanley Park. And then maybe at the entrance, I saw the map and then tried to remember, okay, I hit that road or reached that peak or so. In the middle of hiking, let's say we confronted this crossroad or bifurcating road, and then now I need a map, right? So what I'm trying to illustrate using this example is you constantly, required to retrieve past experiences and align them with the current ongoing perceptions. So in that way, there's many other perspectives to you might take from this example. But to me, as a researcher who's studying perceptual decision making, that past experience can be taken as evidence for decision making. So if you can retrieve more, and then that means you're kind of expanding the time window of sensory evidence. So this is really, to me, crucial for intelligence. And so my question I'm trying to share with you today is discuss how does our brain maintain and utilize those past experiences as evidence to meet various task demands. So this is very seminal work by Bedley and Hitch, published in 1976, Psychology of Learning and Motivation. So the graph you're seeing now is just remembering the list of words, not related words, 16 words, but you have a kind of a preloading task. You have to remember digits, okay? So they try to manipulate the loading, and then you can tell from the sign that you have to more digits to remember, and then your actual performance dropping. But more contemporary type of research, also I'm kind of focusing into a slightly visual working memory, sensory type of working memory. And then now they are showing a bunch of stuffs at the same time or sequentially, and then later queued, and then you have to report. So this is called the reproduction task or delayed estimation task. And then they are kind of looking at the estimation errors. And this is errors and probability. And so it depends on number of items to remember. It's more spread means more probability. And also today I'm going to talk about the mean. So you put together all the trial, it looks like aligned with the physical stimuli, but actually I'm going to show later that there's a certain systematic bias in it. So I want to spend a little bit of time in this very classic work done by Markens, Roma, and Brody, published in 2005 in Science. So the monkey is trying to make a two-interval discrimination task, which is in his left hand, there's a mechanical vibration is a stimuli, and experimenters varied the vibration frequency. So monkey's job is compare this first interval and second interval, which one is higher frequency, okay? So if you just take a look at this task paradigms, kind of bring back the previous example, the Korean hiker at Vancouver and trying to retrieve the map he saw minutes ago. So that's happening in delay period, although this is very brief. But if you look into the sensory area, somatosensory area is S2, there's a completely a lack of activity at all. But you can see in the stimulus period, F1, those two neurons, very nice frequency tuning, but there's no activity at all, seems like. But unlike a sensory cortex, if you move to prefrontal cortex, there is nice, even though there is nothing, but monkey and mind, they kept their frequency. There's nice tuning. These two, the prototype cells are showing completely opposite tuning. So this, my talk is today going back to sensory cortex. I will show you there is something there, not only including my work, but all the researchers demonstrated there is definitely the memory signals in very early sensory cortex. Now moving to I want to stress the content-specific delay activities. What I mean by there is a tuning, right? There's not just the monotonic, the univariate response to the memory load, but it's more the contents of stimuli. So this bunch of contents, location, shape, contrast, orientation, it could be anything. But so far, the monkey electrophysiology, human fMRI found quite similar distributed pattern of neurosignatures of this content-specific delay activity. But looking at the early visual cortex, which I'm going to focus on today, interesting happening. As previously shown by the Roma group, and if you look at the univariate signals, unlike prefrontal cortex, parietal cortex, there is a very minimal activity at all. But if you look at the pattern of activity, there's definitely the signature of the sensory stimuli or the variable of interest. In this case, could be the orientation visual cortex. So there is a early visual cortex. There's a content-specific delay activity there. It's revealed by mostly multivariate analysis. My talk is in two folds, and first is done by Gu-Hyun, currently at Stanford. And I'm going to show you, persuade you that there's interesting attractor dynamics, but also that dynamics is influenced by decision making. So going back to this, I hope you remember this one, the contemporary framework of studying visual working memory, showing and then while varying the load, the number of items, and then we measure the estimation errors. And then we are mostly interested in how spread they are. But I'm going to focus on the mean bias. If you put together all the orientation response, it looks like aligned physical stimuli, but actually, if you spread out the orientation, there's systematic bias in it. And also another canonical bias, I talked about this with John O'Doherty yesterday. And then I also, another series of work on we call it history effect. Our current perception and decision heavily influenced by previous decisions we make. So the same thing here, the estimation, working memory estimation influenced by this choice. We try to remember the memories more consistent with previous choice we make. So clockwise, counterclockwise, actually they are biasing the distribution of estimation. That's two canonical biases I'm going to focus on. So again, it's boring task, but I'm very thankful for a subject who sacrificed their precious time. They come over the fMRI center, I think at least 4 days and 5 days inside the scanner, more than 1 hour, constantly looking at this boring stimuli and then estimating. So I'm trying to here is make it more enjoyable, bring that the hiker guy again. So what we did is show the stimulus after like 18 seconds delay, because hemodynamic delay, we want to get around. So we put the very prolonged delay other than most working memory study. So when then we classic reproduction task, they are rotating the minimized visual information, but they try to match the ongoing perception to what they remembered. So for example, let's say show the real physical orientation like that, and then human estimator estimated, and then there's no bias in case. What about, he rotate a little bit deviated in clockwise, and then in this error plot, you're going to locate this, okay? Another clockwise bias, and then also we can the counterclockwise bias. And so that's how we map this, the estimation bias. So that's why, we make all those, this very classic type, this is conceptual figure, but I'm going to show you real data later, but our subject showed the same pattern. Interestingly, this is cardinal orientation, 0, 90. But the very influencing story is tightly linked to natural statistics. Yeah, I'm not gonna going into details how this natural statistics create this one, but just for my talk, you just remember very systematic stimulus-specific bias exists. The second bias is decision-consistent bias. So again, showing, but in the middle of delay, I gave them the reference, not real line actual experiment, because to minimize the sensory stimulation, we just show 2 diagonal dots, okay, in the fovea. So it's completely different reason from stimuli. So we forced subject to make a decision so that you remember the target was tilted clockwise or counterclockwise compared to reference stimuli. Okay, and then we ask them again at the end of the test, the trial, the estimation, make an estimation again. So so here is what typically happening after making clockwise decision. So you remember, and the judge that you remember past is clockwise tilted, then you tend to, the estimation is deviate away from the physical stimulus in a direction consistent with your decision, previous decision. The opposite thing is same. So we can do this. So that's why we use this plot as a kind of behavioral signature of decision consistent bias. So with these 2 bias, we crafted experimental paradigm. As I stressed before, we created very prolonged delay period, and this is a decision making discrimination. This is the estimation period. We created 2 trial types because the decision making time is unpredictable. So we trial-wise randomly show the early decision-making trial and late decision-making trial. And also again, this reference was very small and occupying different retinotopic locations. So it's very close to fixation and show diagonal dots used in reference. And then the estimation period, the subject rotated using a computer keyboard and reported their remembered orientation. So this is real data. And also I like to emphasize this 50 participants coming to the scanner almost a week. So it's a lot of data. That's why we have a very clean data. This shows a stimulus-specific bias, the reports away from cardinal direction. And there's a decision-consistent bias. So we confirm that the very canonical biases occur in our task. So, and firstly, I show, before going into the results, I also, I really thank the first author, Hyun-woo, who made these animations to make more understandable this seemingly complicated dynamics. But for those who are very familiar with these concepts, you might ignore. So this is just a animation to help you understand the attractive dynamics. So we had two scenarios. Maybe our memory is just not biased, just diffuse, okay? So as function of time. Then this is a typical plot. This x-axis time and y-axis memory states. But in the background, you have little arrows that's indicating the energy landscapes, the slope. So there's no slope, it's flat plane, and then just as time of functions, you just diffuse. So individual lines, you can take it as a single trial trajectory of memory states as a function of time. Okay? So all the same across different stimulus orientation. But another scenario is possible that is diffusion drift. Simply, we can call attractive dynamics. There's not a flat landscape, energy landscape. There is a source and basin. So like a peak and trough, and of course, the memory state tend to drift toward that basin. So in these cases, we have a quite different the phenomenon we expect. So now we have kind of a very diverse. You can see this is a attractor here and here. Just phenomenally to match what we observed, our perception tend to attract oblique orientations. That's why we put it the orientation vectors in that way. So that as time goes by, of course, here is you can see a lot of diverging. Some of them they're heading this way, attractor, and then this attractor. So so for those orientation close to attractor, they actually converging drifting occur. So this is at this point we only provide explanation at phenomenological level. But anyway, so this sums up to possible dynamics scenario, what might happen during delay period in memory states. Then I try to relate the predict based on these two scenarios. First, to start with diffusion-only dynamics. And although the variability increasing over time, but if you take the mean, there's no bias grows as a function of time. So early period and late period, taking the mean, there's not much change. But here's the different cases. And then early is a small stimulus-specific bias. It's predicted, but later is the more growing degree of bias is expected. So that's the two different predictions we are testing. So this is, we ran ex ante simulation, just based on a typical range of dynamics. And then this is expected from scenario one, diffusion only dynamics. And this is the attractor dynamics. So attractor dynamics only predict increase in stimulus-specific bias as function of time. So this is all model ex ante simulation. And then this is what we found in human subjects. Still, we're talking about behavioral data. And behavioral data, although small, is a significant increase. By the way, this phenomenon was also shared with Dr. Kongtae. He shared some of data he recently presented at some conference, and he replicated this phenomenon. Again, so in behaviorally, it supports the second scenario. There is the attractive dynamics is going on. And then move on to the decision consistent bias predictions. So the same, but we make a decision here and here, right? So it slightly different dynamics made by these two scenarios. So again, the diffusion only dynamics. So happen to be the diffuse in this side, so subjects make a clockwise this is making, right? And then we assume that, that decision kind of shifted that initial state toward and direction consistent with the previous decision, okay? And then in that case, you focus on this after that. This is totally just a statistical dependence. Happen to be that side, happen to be that side, but after that, it's just diffusing, okay? So if you take the mean, there's no increase in decision consistent bias. So early and late, very similar, but things are a little bit different in the second scenario. Now, I think already there is a constant bias. This will influence your upcoming decision making, right? And that decision making also add another bias. And then after that, keep continuing toward that same attractors. So this dynamics has different after making decisions, they keep growing. And I'm not gonna show the other stimulus case, converging case, but diverging cases are there, keep increasing this decision consistent bias. So I just sum up these two cases, make a two difference. It is more schematic illustration of mean bias. So using these predictions, we put in the phenomenological model, and then we run the model fitting and then definitive model. All 50 individuals are without exception, although very small for some individuals, of course, but supported the second scenario. So sum up this all behavioral phenomenological level analysis supported the second scenario. There's attractor scenario better explain stimulus-specific bias growth, and this is inconsistent bias growth. So then I'll move into fMRI. So we used pretty conventional classic inverted encoding model, this kind of MBPA, but more robustly confirmed by many several groups. So we adopted that technique. So this is a simple, again, they bring up the scenario 1 and 2. And now they predicting that the converging bias toward attractors. So if you measure that, as time goes by, and this stimulus-specific bias will keep growing either early and late trial, although there little difference in dynamics. So the same again, again, the all behavioral data were collected inside the fMRI scanner. So at the same time, we look into early visual cortex pattern of activity. And then this is the one single trial. Of course, we put a lot of trials together and average. That's why we have such nice and smooth trajectory of memory states. So if you put all together, the fMRI data actually shows the same pattern that toward oblique orientations, they're the converging, and then push away from the cardinal orientation directions. Early and late trials show the same pattern. So in a way, the fMRI data supported again, the stimulus-specific bias grows over time. And then also the same, the decision consistent bias, the early trial, later trial. The phenomenon you see here is actually model predicted at the presence of reference stimuli, they tend to attract the memory. So this is kind of matched with the model prediction. But important thing is there, the matched with the drifting dynamics idea. So we run the model and confirm that the majority of subjects are supported the drifted diffusion dynamics, attractor dynamics. In part 3, so this is hard part. So we, because so far we're talking about the phenomenal level, the modeling, and also fMRI, but we really wanted to know what's the neural mechanism, mechanistic explanation for why we have such the attractor dynamics was influenced by previous decision making. We wanted to understand this phenomena at the mechanistic level. So we are not doing electrophysiology monkey. So we're just using artificial neural network, just seeking a possible potential neural mechanism that enables this phenomenon. So this is basic architectures RNN. Of course, real artificial neurons are a lot more than this schematic the capture of the RNN model. But two things, you see two different the group of neurons. We assume that because we should one represent the past experience, the target stimuli, the gobble patches and gratings subject saw before at the beginning of trial. But also you also have another input is reference. Okay? So this just visually show single trial artificial neural activity, and we show the stimulus show up here and reference show up here, and then you have a raster plot. Okay? So it's just they make a decision here. So that's why the reference stimulus lined up here. And also this, after training, RNN network show there's the bump-like attractor dynamics. So this particular set of neurons show higher activity. They keep maintaining throughout the time. Okay? And then also they also couple those different group of neurons, actually reference-taking neurons, they show a signature of decision-making. There's a winner-take-all behavior during the decision-making time. But an important thing I want to stress in this recurrent neural network modeling, we did a joint cost as a function of both estimate and decision making. So this neural network's goal is make as precise as possible estimate and also decision making. So we trained for both type of cost. So then, of course, we had to make sure that they capture the human behavior. So they demonstrated nicely the stimulus-specific bias, also the decision-consistent bias, and also fMRI data they replicated. I previously mentioned that this little dropping phenomenon also predicted by phenomenal model, but also RNN showed that it's the same patterns. Okay. But our real interest is the understanding. So they just passed the criteria. Okay, we can recapture human behavior and fMRI activity at phenomenal level with this trained, task-optimized RNNs. But what we're really interested in the mechanism to drive this phenomenon. So we're closely looking at, we pretend to be electrophysiology on our artificial neural network. So we try to make a raster plot and then draw it. Looks complicated, but in a nutshell, I just using these panels and explain what's happening. Its mechanism initially show the stimulus is aligned to 0 around, and then reference show up these two reference tuned neurons. One is clockwise and counterclockwise. They're kind of competing, okay? But there's a winning group of neurons. They are winning and losing. But what's happening in the memory population of neurons, they are attracted toward the winning populations. So this is the description of activity space analysis. We can move on to state space analysis. We can geometry. This is low dimensional state space. We can, this is well-known mechanism, the addition rotation, which was found in the flight system too. So navigating at the stimulus, they are changing their heading directions. We were really surprised to see the same mechanism going on. So decision actually perturbing, and then the memory systems throughout the breaking of symmetry. As a result, the related memory systems are the steered toward the decision consistent directions. going to skip. So the summary of part one is we found basically in early visual cortex doing fine neuroimaging and combined with the phenomenological level model and then artificial neural network model, we demonstrated that there is a memory content specific memory information early visual cortex, and even though match it with the very well-known canonical biases, and then our artificial neural network propose possible explanation at mechanistic level. I move to the part two is really related to the first one because we use the same data. This was done by Sungjae is here. Yeah, he's sitting there. So these all figures are made by him. I just edited add some words. So that is this tentative title of the manuscript he and I working on. So we gonna claim that, so visual cortex is known to represent incoming stimuli, right? That's what sensory cortex is known to do. But on the other hand, we found a very robust working memory signal. Then there's someone might question that, don't they interfere with each other? Okay, they might create problems. So idea is they might be represented by different codes. Okay? That idea is found actually premotor cortex recently a lot, the low dimension reduction study in binding subspace. But I just, to anticipate the conclusion, we believe that they are residing in almost orthogonal subspaces. So this is the, quite a series of work adopting delayed estimation task, and they are looking into early visual cortex for the signature of the working memory. But we found this is so they use the pattern of activity right after stimulus, most of the studies, except a few, only one or two. But I think this is, of course, they did a great job for their purpose, but for our purpose, we cannot separate the sensory response and then delayed memory response. This is not problem at all at electrophysiology. There's a very fine temporal resolution, but board is a sluggish response. So that's why we had to introduce sufficient delay. So that's the kind of nice feature of our paradigm. So again, the same paradigm, the same data, but we have enough time of delay. Another thing is we try to use the techniques that had been used in electrophysiology. So we use kind of a state analysis a lot. It's a huge number of neurons, so people adopt the dimensionality reduction technique. But the classic PCA has a problem. It doesn't care about task structure at all. It's just the data maximize variance explainability. But thanks to this group, they introduced demixed PCA. But it's an amazingly simple idea that, rather than taking just response, they conditioned on the task structures, task vectors. In our case, stimulus orientation. So in that, unlike regular PCA, this is task component. They're all mixed, but this allows us to demix. Let's say this is time or stimulus or strengths, whatever task factors. You just average and using that and then carry out principal component analysis using the same principle. This is, again, the prediction. So there's two scenarios. One is, okay, this is sensory code. We just taking voxel, pretending they are neurons, and they have orientation tuning. Okay, so you have just a very three different neurons. They can create three-dimensional state space. They can embed the manifold, orientation manifold. let's say one scenario is memory code. What if they have the same code, but just the reduced activity, right? We know that the univariate signal is quite low from fMRI study and Roma groups study. And then they maybe shrink in size and a little bit dislocated by the same plane. You know, to make long story short, the meaning of the being together on the same plane is sharing the same linear weight kernel. Okay, they just using decoded by same weights. But what if they have a totally different tuning, and then they will reside in different subspaces? That's the idea. So we carried out, and then we computed the angles, and then that angles, it's very complicated to explain, but this angle is almost close to 90 degrees. And also, if you project to each other's plane, the area is quite shrinked. So that's why, but if you pay attention to this is a sensory stimulus period, and this is a delay period. So we have a nice plateau of the delay-related activity, and it's really stabilized on this plane. So which means the code can be decoded using the same weight vectors. And then we move on to another analysis we carried out. This is bring back one of the old slides that I showed before. In the first part, we apply this inverted encoding model, and then decoded orientation signal. This is what happened. You know, this axis shows the fidelity. That means the inverse of errors. So higher values means decoded well. Okay? So this one is trained on the early phase of trial. That means, you know, where the univariate signal showed the peak response. So we will be stimulus-related response. They trained on that. So of course, train the period that they will very high fidelity, but soon dropped in delay period. Totally opposite thing is happening in delay period. We only trained on using this period. And also this is cross trial types. We trained only early trial only. So we never trained for late decision-making trials. Although early late showed pretty stable decoding fidelity. So this is a temporal separations. It's just a visualization of this decoding results. So second, cross-coding fails between sensory and memory decoded signals. And then also we inspected individual voxel analysis. And individual voxels, we can characterize individual voxels, what orientation they like. Okay? So this shows that, so this is the voxel orientation preference we found in the stimulus sensory period. So that's axis sorted out in that way. And then of course, this trivial, right? So we put the sensory period and stimulus orientation they liked, and the nice diagonal lines. Okay, this must be aligned. But more interesting one is how about aligning them in the late period response? That's completely gone. Now, so there's no match between orientation preference. The opposite thing happening in when we train the late period of signal on stimulus and sorted that way, exactly the same data, but the structure is completely gone. So this means individual voxel level orientation preference is very inconsistent between sensory and memory representation. Then we carried out another analysis. The one of our staff scientists at the center, Ju-Hyung Ryu and I, published last year. And then this is just part of the papers in early visual cortex. There's also bias. There's radial bias. That means this is retinotop, you're fixating here. So the upper visual and radial 45, this is a location, right? I just illustrating their typical orientation preference. is matched to radial position. That means horizontal meridian, there are more neurons with horizontal orientation preference and vertical orientation preference. That actually predicts if we sort out, the stimulus angle and then the angular position on the axis, so retinotopy radial angle, and the y-axis orientation preference. It is predicted that the diagonal line, okay? So this indicating the radial bias, is well-known bias, is only observed in the early stimulus period, sensory period, but nothing like that. It only shows the bias, nothing to do with the radial positions. So again, we show that another distinction between sensory and memory, radial bias exists only in sensory. And then next couple of slides is very complicated to my best, but I really want to show this is, to me, is most important slide because we now, so far, we keep claiming that to sensory representation, memory representation seems to have distinct codes, okay? But what if they coexist? Like a hiker guy, the retrieving the working memory back and compared with the ongoing sensory perception, okay? Actually, that's what's happening in decision-making period because at the center, you have incoming stimuli, okay? And also, although there's nothing on the screen, but in your head, you have a working memory representation, right? So we concentrated on this decision-making period. And then this y-axis is, we actually show different kinds of orientation, but we actively align the true orientation axis. So this is the sensory code readout, and this is memory code readout. So horizontal lines, and then x-axis, we put the reference orientation. So reference is a sensory input. So if they respond to sensory input, they show this diagonal line, okay? But in memory period, they have it flat. That means they don't show any response to sensory stimuli, even though they are vividly present in the screen. So that means this is important, I think, because this is actual demonstration when actually sensory input coming in, but still there is a separately read out, can be read out, memory information and sensory information. And another, also we can use estimation window because we show randomly starting position of estimation dot. That also carries the stimulus information, right? So we divided the clockwise position started trial and counterclockwise trial. And again, that difference only show up in sensory decoded orientation, but not memory. Memory just to show the same response. That means they don't react to, don't carry any signal of a stimulus. And so I show the attractive dynamics, and also the memory code showed the oblique orientation bias, but sensory is the cardinal bias. So this completely different bias. So again, memory matched the perceived bias. And also this is inconsistent bias. I'm not gonna go into detail, but the sensory code, there's no difference between clockwise choice and counterclockwise choice, but only memory decoded one show the neurosignature of decision making. This is inconsistent bias. So let me sum up the part 2. And so we claim that distinct coding for sensory and memory representation in human early visual cortex. I'm going to skip all the supporting evidence, but also I want to finish summary with there is a signature of choice only in memory states. The reason I stress that conclusion, I'll show you in the next slide. But so this is takeaway. We just concentrate on this one because this project is started with the question that the audiovisual cortex supposed to represent sensory signal. Is it nice to have a memory signal there? Okay, might be interfering. There's a lot of people worried about and criticize. There's maybe artifacts or just the spurious signal that's not, contributing real causal distribution to maintaining memory. But I cannot, to counter argue, but our data suggesting there is a possibility that they coexist happily and peacefully without interfering each other. That's maybe something in the memory representation. Songjae and I suspicion that might reflect the top-down signals. So yesterday, Dr. Fukunaga shared with us layer-specific imaging. So that's why the Songjae traveled all the way to the NIH, and we started collaboration with Eli Merriam for layer-specific imaging. The idea is in audiovisual cortex, there is a memory signal, is more the feedback layers. And this is my last slide. I mentioned that, the choice-related signal finally in audiovisual cortex, I mentioned the finally, because since I arrived at SNU, I occasionally visit audiovisual cortex several times. I kept failing. They're very reliable. They're picking up very minimal difference in sensory signals, but it's hard to find the choice-related signal. And also John's talk and all the visual features, the hierarchy of value, the information accrual, the same thing. We keep searching or kind of end up with a little bit disappointing results, but just the memory-related signal finally show up in all the visual cortex. But paradoxically, there was not, the period where activity hit the highest mark, but actually it looks silent, but you're looking at the pattern of activity that carries really robust, nice memory, but also reflect decision making. So thank again for Hyunwoo, Gu-Hyunwoo, and Kim Sung-jae, but also I heavily indebted to development recurrent neural network was collaboration with Dr. Lim Suk-bin at NYU Shanghai, and these are our lab members. Okay, thank you.
Thank you, Professor Lee, for the great talk. Got time for questions, John?
Really very, very interesting. Yeah, I was just wondering about the recurrent neural network result. And what I was wondering is, obviously the fact that it is recurrent and it presumably has attractor states would be the reason why you're seeing kind of similar effects when it's trained. But I'm just wondering, is there any other architectural aspects of it that would make it show the effects? And are there any regimes or any architectures that could still maybe do the task, but not show the same kind of dynamics and the same properties that you're seeing in the, humans are exhibiting?
Yeah, that's very important and critical question. The one thing I should mention that this is a pretty simple recurrent neural network. The vanilla network, pretty simple. Hyunwoo and I and Professor Im Suk-bin, we started with just single neural population at the beginning, but we couldn't find the neural signatures, and then we discussed a lot, and then we realized our paradigm actually separate input in different location of retinotopic input. So we might, how about create two different populations? But another emerging group of neuron is actually the clockwise preferred neuron and counterclockwise preferred neuron that just naturally showed up. And another thing I want to bring up, I just skipped for the time pressure, but we show that the stimulus-specific bias, the constantly drifting toward a few attractors. So we started with this homogeneous the input, but we couldn't see that stimulus-specific bias. What we had to do is actually the mimicking the input in the environment. We show more the cardinal directions, cardinal orientation. Now we begin to see this ellipsoidal phenomenon. So to sum up, to answer your question, we started with just single population, but didn't work. So we separate to the memory population and then the reference sensory population. And then also we change the stimulus input match to the state environmental statistics. Then we began to replicate all the phenomenon we saw that drifting dynamics, and this is inconsistent bias. But also another last thing I want to emphasize is I mentioned that we trained the joint cost, right? And then one trial, we just removed the decision making, and then didn't show up any such activity. So that's why we, in our paper, we conclude this is very necessary, including the decision cost. And actually the beneficiation of that thing is making decision making more robust in the presence of high level of noise. So that's our takeaway.
Thank you. Thank you for explaining clearly and simply content that I really don't know much about. I think there were elements of perception in there and decision making and working memory. So it was quite a lot to take in. But as somebody who studies memory, for me, episodic memory and not so much working memory, but I'm always having to deal with the kind of mystery of how it is that when we reactivate memories, we actually have some kind of, we see these signals in, in our fMRI results of the visual cortex always being involved in our memory reactivations, even though you're not looking at what you saw before. How generalizable do you think these kinds of principles are to the function of the visual cortex? And I guess that's one question. And a little bit deeper than that, how the heck does the brain manage to simultaneously hold these two things in parallel? Because whenever, if I ask you, remember this morning, you're definitely looking at stuff, but you're also activating these other perceptual representations from before. What kind of mechanism do you think is handling that kind of amazing ability?
That's great and difficult to answer your question. Really important question. The first part, actually, I just talk about this future experiment, which actually got started already, layer-specific imaging. So we believe that this something related to top-down signal. But also another direction I think we should go is not stay in early visual cortex. I think I want to generalize this phenomenon to other, like a face area or other motion area, the same situation. You're dealing with just the color actually look better represented in V4 and V01, a little bit high tier cortex. So there's also color manifold nicely can be imaged by fMRI, quantitative fMRI. But I would predict, if my claim is correct, they also, there's a feedback signal. We can read out the distinct neural codes. That might be general. That actually related to next question. How is it possible? So I cannot answer that question right now, but in our field, there's lots of debate, but this is sensory recruitment hypothesis. So the people against that idea is that exactly that point. Lots of interference, but at least our work shows that don't worry about. There is already there are a bunch of studies showing the separate, the motion planning area, premotor. There is action space and also planning space. So a bunch of examples, they show up. So at least we just opened the door, and then I wish we can do the test of the same idea in different parts of sensory cortex.
Do you think that there are like mechanisms that could be tested in animal models? I mean, of course, there's a limitation to fMRI, but in order to kind of get to the mechanism, like
yeah,
how
that's
the
also that's why we did RNN, and then I'm not gonna do it, but
somebody can do it.
electrophysiologists, please try, test our prediction.
Yep, thank you.
Okay, any other questions? Please.
Thank you for the inspiring talk. You introduced us about two studies. The first one about the drift diffusion dynamics of memory, and the second one about the separation of sensory and memory code. My question is that how do you think these two can connect? Like for the sensory and memory code to be separate, how do you think that during a decision-making process that the drift diffusion dynamics interacts with sensory code?
Yeah, it's good question. And just because of time limit, I skipped the slide. But actually one result Sungjae created is that actually they're looking into relationship between sensory representation, memory representation, their errors, decoded errors on a trial-to-trial basis. Although pretty weak, there's a little bit of a correlation. So sensory cortex in this trial happened to be biased toward clockwise. And we see the same tendency in the memory code in the same trial. So that's important because orthogonality does not mean not linked. They just separately represented. But of course, the memory is originated from the sensory input. So although lots of noise process coming in the causal chain, the feed forward and feedback chain, but still there's a relationship. But it's although weak, we see a significant correlation. So we can conclude that they are separate, reside in separate subspace, but they somehow correlated. Again, this similar thing is reported in the premotor cortex. This planning and actual action is related, right? So they are orthogonal subspace reside, but they are correlated. So orthogonality is association is completely compatible with each other.
Thank you.
Do we have any questions from online? John?
Yeah, I wanted to, given that this is a neuroimaging symposium, I wanted to ask you a question. It's kind of more general question. So you've kind of been doing this kind of decoding analysis and demixed PCA, and these are things that have been done at the kind of single neuron level with looking at neuronal populations. But obviously you're looking at voxels, right? And this is of course a long tradition of kind of using, which I've done and with which you're doing, of doing these kinds of decoding type analyses and looking at the information representation using these techniques in fMRI voxels. And I just wondered what you think about why does it work, right? Because of course an electrophysiologist would say, well, the voxels should have no information because you're averaging over hundreds of thousands of neurons. What do you think is the nature of the information that's preserved as you go across spatial scales? I don't have an answer to it. I have thoughts, but I just wondered what your kind of general thought is, and whether
Yeah, it's thanks for coming up with this important question. So this is not necessarily you know, pertaining to my particular talk, but in more general, in a sense. So I want to talk about the figures showing up in right upper corner. So this is kind of Frank Tong's vision. There's a famous debate, but I think the mixed stories might be true. So his idea is, if you look at really, there's an orientation wheel, right? In the monkey electrophysiology, show that structure. And also the RICAN group showed actually high resolution neuroimaging show. So that's what's happening at the single neural level. So we call it this orientation wheel. So it's column structure that is far more smaller than fMRI voxel size. But Frank Tong claims that that still reflects bias. So that are picking up. So that's why the inverted encoding model actually utilized that idea that coming up with assuming that there is a bunch of tunings. This is a latent tuning curve. And then each tuning curve has a more relationship with individual voxels. But that's one story. Another story is they're related to experimental design. I already talked about the radial bias. So some people, other group, including Eli Mariam, there's definitely big factors that the radial bias they are picking up is radial bias, right? Because the retrotopic location is pretty, big signals, and that correlate with the orientation. Okay? So that could be so that's the maybe other stories possible, but I want to bring up one of the slides here. We're actually looking at the radial bias. So that actually true. The sensory period is definitely we observed several times in our previous work. And we see that again. But here, there's no such a thing at all. So at least our memory representation, there's no such a thing as radial bias at all, but we reliably read out the orientation signal. So there is no, the black and white answer, but still our data shows the Frank Tong's story is partially true. Yeah. So even without global scale bias, we can still read that orientation information.
Although it's definitely side of primary visual cortex, you're not going to have those, as far as we know, like in prefrontal cortex, this type of columnar organization, but yet we can still decode,
Yeah, that's a very important part, because the reason we tried the parietal cortex, prefrontal cortex, several other groups have tried, is typically it's hard to find, not because they are not signal there, but just it doesn't fit with the fMRI. Because all the visual cortex, they have nice functional architecture governed by orientation, right? But if you move up in the hierarchy and you have not such nice geometry, so doesn't fit with this technique. So that actually tells us important the if then the statement. If you found some signal that indicate there's some information related to, okay, regardless of functional contribution, but if not, we cannot conclude there's no signal there, because it is still possible that just the MBPA technique cannot pick up the information because very limited and then requires a certain set of assumptions. Yeah.
Any more questions?
You may conclude.
Okay, so before we end our symposium, Professor Lee, do you have any final words to share?
Yeah, of course I have. I want to turn off my phone. Maybe we show the symposium, the website. I already, at the beginning of today's sessions, I kind of briefly reviewed the yesterday's speakers, and I also, these two speakers just left because the Dr. Nakajima have a flight to catch, so they are running out of time. So there are a lot of questions about this two talks. And so one of the reasons that we purchased a scanner, and then we can use for basic research and pure cognitive neuroscience questions, and also either brain structure, but also one of the reasons we got awarded for this grant, which is huge money, but part of the reason is that we can do something to benefit human quality of life. So although I know that the clinical neuroscience is a very difficult thing to do, unlike what I do. I live a very simple world, right? Just the showing stick and estimate orientation, well controlled, but it's kind of hard to relate that to real world phenomena. There's many things and factors to explain. So these two scientists in collaboration, struggling to help the suffering minds, schizophrenia, and then modern society, the increasing the morbidity rate in the psychiatric issues. And also we also the pair of talks and led by John O'Doherty and Woo-Young An. And I'm personally doing our perceptual decision making. So perceptual decision making is actually about making inference based on evidence, sensory evidence, and inferring the state of the world, kind of objective world. But I found that, I just prompted to ask you questions, but, I feel like when you're answering one of the questions from audience that what about artwork, this how the context dependent. I think you gave a beautiful answer for it, but also I was sitting down thinking of that. Oh, there is something that the objective value there, right? The price, but also I have subjective value, and that creates gap. And then that's kind of creating meta value. Okay, we call it Korean 가성비. We pay little and then getting more. So I think that's somehow related to that. So that's another interesting direction. So the reason I'm talking about that, my personal take is that that's the purpose of this symposium. I mean, although I'm not studying value-based decision making, but while listening to your talk and Woo-Young's talk, it opened up my mind to that direction and possible interesting collaborative work. And again, sorry for missing him. It is his face. And but next week I visit him at the conference, and also we have the private visit. So we going to continue to collaborate. And Dr. Fukunaga yesterday, the NIPS program has also nice layer specific imaging. So maybe in possible future, we might collaborate in that direction too. So I already collected some of the data. We haven't analyzed yet, but some interesting stuff show up, and then I might contact you. Okay. So as the director of the center, and also this responsible for organizing this two-day international symposium, I'm really thankful for the esteemed speakers who found their time to make long visit. So I know you're a very busy schedule, but your presence means a lot to us, and then give a kind of a jump start and then realistic feeling of getting started. So I really felt yesterday and today, in addition to the official ceremony. Yeah. And also I'd like to thank Dr. Ryu and Im Jae-seop. I have a memory problem, yes. So other staff scientist, Yun-sang 교수님, I want to pronounce. I don't have glasses now. And also other staff members and all the students to show up in the middle of the semester. Let's give big hands to ourselves. So this is official end of symposium. Thank you.